Oh, my hair looks horrible.
Mine too.
So sorry, I'm not muted.
That's all right. Mine too. All right, let's go ahead and get started. Today is Thursday, July 8th. Again, just like last time, lecture will be muscle physiology, and then we'll focus on the origins, insertions, and actions for lab. Again, 4 more assignments to do over the next 3 days. So you have all of that there. Remember, there's 7 different lab reports you're doing for the PhysioX. Remember, when you go to print it, select the print, print option, and just save it as a PDF. And then remember, also your muscle activity handout is graded for correctness. And then Thursday, 1 week from today, you have your lab and lecture exams. So again, busy, busy, busy. So let's go ahead and get started. Any questions on the schedule before we get going? All righty, excellent. Then let's go back to the lecture. We have been working our way, and actually, let's go back to the whiteboard. We have been working our way through a muscle contraction. Excellent. And last class, we did part 1 of that muscle contraction. And what was part 1 called again?
Initiation of communication at the neuromuscular junction.
Sure, absolutely, absolutely. That's a perfect way, or we also said communication. That's fine. Excellent, communication at the neuromuscular junction. And remind me again, what was the goal of part 1?
Convert neural action potential to muscle action potential.
And I'll just abbreviate it because I think we've written it enough times for right now. Plus, it's not the point we're trying to emphasize. All right, let's add to this, though. What was the first step in that process?
Making a decision in the brain to move the muscle.
Excellent, right? Because we voluntarily control our skeletal muscle. So the first step was making a decision. And what was the last step?
The muscle action potential down the sarcolemma.
Excellent, I like that. We produced a muscle action potential. I'm going to expand on what you said, but you're absolutely correct. We produced a muscle action potential at the motor end plate, and the muscle action potential spread down the sarcolemma. And make a point of emphasizing that, because now that we've done part 1, we need to move on to part 2. And what is part 2 of our muscle contraction process?
Excitation, contraction, coupling.
Excitation, contraction, coupling. Excellent. What is our goal of step 2?
To move the regulatory proteins.
Excellent. Excellent. And if we wanted to cheat a little bit, we could add one little thing. How are we going to move the regulatory proteins?
Release calcium.
Yeah, we're going to use calcium. Again, you're not going to have to write the goals. I write this for me because it helps me to remember. And now let's add one more part. What do you think the first step of excitation, contraction, coupling is going to be?
Getting the muscle action potential down to the reticulum.
True, but even before that, what has to happen before that?
Calcium triggers movement of myosin.
Well, that is what we want. That is our last step. That is going to be our goal. But as you guys have planned, remember, as we've talked about, this muscle contraction is a continuous process. So the end of the first step is also the beginning of the second step. So our first step in excitation, contraction, coupling is
muscle action potential spread down the sarcolemma.
Is produced at the motor end plate, and the muscle action potential spreads down the sarcolemma. There you go. So that's the point I want to make. Because this is a continuous process, remember what is the last step of part 1 is also going to be the first step of part 2. And you're right, our last step, even though we haven't gotten to it yet, is going to basically be like our goal, move those regulatory proteins so myosin and actin can interact. So that is our game plan. All right, so let's take that, and I'll cheat and copy that so that I can erase that. Put that there. Excellent. Now, just like we did with part 1, we need to start with the anatomy. So let's do some drawing. As we know, we are going to start, at the motor end plate. So let's make our little squiggly motor end plate right there. In fact, we'll put the whole neuromuscular junction there. All right. Oops. Wow. There we go. So we have our neuromuscular junction. We have the rest of our sarcolemma, which we know is specialized to produce at the motor end plate and propagate a muscle action potential. But we also know that there are invaginations of our sarcolemma. And what are those invaginations called again?
Transverse tubules.
Excellent. So we know we got those transverse tubules. Those transverse tubules are one of the 3 components that form a triad. What is a triad again?
It is the transverse tubules and the 2 ends of the reticulum thing.
Sarcoplasmic reticulum, excellent. And you are right, we have this web-like sarcoplasmic reticulum with these big enlargements at the end of it. And what are those big enlargements of the sarcoplasmic reticulum called again?
Terminal cisternae.
There you go. Well, exactly, the terminal cisternae. Excellent. So we have that. This is all stuff we know. We also know that the transverse tubule and the sarcoplasmic reticulum wrap around our myofibrils, the contractile organelles. And what was the organizing structure of a myofibril again? Myofibril is the contractile organelle made up of a bunch of repetitive boxcars of proteins. What was that boxcar called again?
Sarcomere.
Sarcomere, absolutely. What are the boundaries of the sarcomere?
Like the A line, the I line.
Well, true, but what are the walls? What are the boundaries that define a sarcomere?
Z line.
The Z line, made up of that protein that is the Z disc. So we know there's a whole big long myofibril here, but I'm just going to draw one sarcomere here because for this step, it will serve our purposes. That, of course, has a Z disc. That, of course, has our actin filaments. And I'm just going to draw a couple of them. And then we also know that thick myosin filament is here. That has those heads that want to interact with our actin. And I guess I need to cheat here a little bit. Let's go ahead and make my actin a little bit thicker. Again, it's not a thick filament. It is one of our thin filaments, but I want to make sure we distinguish it. Only the myosin is the thick filament. But I wanted to do that because then that allows me to put my thread-like regulatory protein And what was the thread-like regulatory protein called again?
Tropomyosin.
Tropomyosin. And tropomyosin has these little handles on it. The troponin. Excellent. All right. Again, there's actually no new information here, but let's go ahead and label everything real quickly, just again to go through it, make sure we're comfortable with all of this. This, of course, is our neuromuscular junction, and we'll just abbreviate that here. This is our sarcolemma. This is our transverse tubule. Remember, once you've written out transverse tubule once on a essay question, you can then abbreviate it T-tubule. But if I ask you to identify this on a lab exam, make sure you spell it out. Then we have our terminal cisterna. That there, which is in the large portion of our sarcoplasmic reticulum. And then let's go ahead and really small remind ourselves of all these proteins. Our Z disc, our myosin, our actin, our troponin, and our tropomyosin. I think that's everything. All right. And that's all the stuff that we know. However, let's start adding a couple quick things. For starters, what is the function of the sarcoplasmic reticulum in our muscle cell?
How's calcium?
All right, it stores and releases calcium. Let's start with the storing part first, right? As we know, calcium makes cells do wonky things, kind of like criminals do wonky things. One of the places where there's the highest concentration of criminals in the Northern California region is Folsom, California. Does that mean you don't want to live in Folsom, California because there's so many criminals in Folsom, California?
They're locked up.
Exactly. They're locked up where they can't cause any wonky things to happen. And the same thing's here. Calcium makes a cell do wonky things, and so we want to keep it locked up here. One of the ways, actually two of the ways we do that is, as we know, there are calcium pumps, and we are going to have some of those calcium pumps. Calcium pumps, of course, as we know, use ATP, and they use that ATP to move calcium into the sarcoplasmic reticulum. And remember, we also talked about outside of the matrix of bone, we have the highest level of calcium here inside these sarcoplasmic reticulums of our body. And so we're able to do that because, among other things, there's a special protein here called calsequestrant. Again, it's one of those funky terms that you're probably going to have to spell at some point. But again, the Ca refers to the calcium. Sequestrant, what does it mean to sequester something?
Bring it in for questioning.
Well, okay, it is bringing it in, but it isn't so much the bad guys that they sequester, although I guess they do that sometimes too. I always think in terms of the juries, right? If you have a jury in a really politically, a fiery type of case, they don't want them listening to the news, reading the newspapers, being on social media. So they take those jurors and they lock them away so that they can't be exposed to those outside pressures and things along those lines. So we have this special protein in here, the calcium pumps. I don't think I actually wrote calcium pump here. to move those in there and to lock the calcium away. Right? Just like Folsom Prison, we want them locked away where they can't cause problems. But this is the excitation contraction coupling where we want to use calcium to move those regulatory proteins. So that means we need a way to get this calcium out of the sarcoplasmic reticulum. And luckily for us, such a thing exists. In fact, we've already seen it in the previous, I guess I should put a pump over here as well. In the previous process, we had a gated calcium channel. And what was the key that unlocked that calcium channel?
Voltage gated.
Go. Just like we saw before, we have voltage gated calcium channels. And of course, when these channels open, which way is calcium going to flow? Out of the sarcoplasmic reticulum or into the sarcoplasmic reticulum?
Out.
Out. So exactly, when open, calcium leaves the sarcoplasmic reticulum into the cytosol. Excellent. So with that, we now have the anatomy that is going to help us to understand part 2, excitation, contraction, coupling. Questions on this? Again, as we've talked before, if this doesn't make sense, it only gets worse from here. So let's make sure we understand before we move forward. Excellent. All right. Then just like last time, let's switch gears now and talk about initiation of excitation, contraction, coupling, right? That's the start process. As we already know, where and how does excitation, contraction, coupling begin?
When the muscle action potential is produced at the motor end plate and spreads down the sarcomere.
Exactly. Does that mean that if I ask you on the exam to describe initiation of excitation, contraction, coupling, you've got to go all the way back to the beginning of making the decision and producing the neural action potential and all of that? No, absolutely not. You just start with production of a muscle action potential at the motor end plate that spreads down the sarcolemma. Of course, as we know, muscle action potentials, just like neural action potentials, are a big positive signal. And as we know, when a big positive signal spreads, what does it do to the area that it spreads to?
Depolarizes.
Excellent. So as it spreads, it depolarizes the cell. And sure enough, this muscle action potential is going to spread from the motor end plate. Nope, hold on. I want that to be blue. From the motor end plate down the sarcolemma. But guess what? It's also going to spread down the transverse tubule as well. So our muscle action potential spreads down the transverse tubule. And again, this is a big positive wave. So this big positive wave spreads down the transverse tubule. It depolarizes the entire triad. of the transverse tubule and the two terminal cisternae. Now again, because we're always thinking of the immediate effects, what is the immediate effect of the depolarization of the terminal cisternae?
The voltage-gated calcium channels opening.
There you go. Voltage-gated calcium channels open. Excellent. And when those voltage-gated calcium channels open, what happens next?
Calcium makes a run for it.
Exactly. I like that. Absolutely. We have our jailbreak. Calcium is now, okay, those are too big now. Calcium is going to be released from our sarcoplasmic reticulum. And as a result of that, we get an increase in calcium levels in the cytosol. Notice the overall amount of calcium in the cell hasn't changed. Its location has changed. We have our jailbreak. It is out of the sarcoplasmic reticulum where it's locked away into the cytosol where it can make wonky things happen. And in this case, when released from the sarcoplasmic reticulum, remember the sarcoplasmic reticulum is sitting right on top of the myofibril. So all that calcium, as it comes out, is right here on top of the myofibril, where our calcium can now bind to the troponin. And here's one of those phrases that we love so much. When the calcium binds to the troponin, troponin undergoes a conformational change, which is a really fun way of saying what again?
Changes shape.
Yeah. Just a fancy way of saying that it changes its shape. Perfect. Excellent. So our calcium binds to the troponin. And when it does that, our troponin changes its shape. And when troponin changes its shape, it rotates. And when it rotates, basically it turns the tropomyosin. So basically what happens here is that when the troponin changes the shape, the tropomyosin is rotated off of the binding site. So let's go ahead and grab that one, move it there, grab that, move it there, cheat and do that. It's something I can't do on the whiteboard, so that's always fun. It rotates it out of the way. And now that the tropomyosin is out of the way, so now our actin, pardon me, let's rephrase that. Our myosin can now bind to the actin and start the contraction. And there you go. We've accomplished our goal. I warned you step 2 and 3 wouldn't be as elaborate as step 1 was.
Now, earlier we were talking about this being a theory. Is it a theory because it's not like
No,
really?
sliding filament is a theory. The sliding filament is the part that's a theory. That's the contractile cycle that we're going to talk about. And again, it's a theory Because again, we are talking about proteins that are actually moving in relation to each other inside of a cell. While we can observe these proteins as we've seen with an electron microscope, you can't use an electron microscope on living tissue because you're bombarding it with electrons and the tissue is actually destroyed in that process. So we've never actually seen this take place. But have we looked at muscle tissue when it was relaxed and muscle tissue when it's contracted and looked at the differences and seen that there's more of an overlap and all those types of things? Yes. So again, technically from a scientific standpoint, the contractile cycle is a theory, but gravity is also a theory as well. And I don't think any of you are going to be jumping out of your second story windows just because gravity is a theory, right? So it's the same thing here. It's scientifically it's a theory, but it's a pretty strong one. All right, but again, that's step 3. We haven't gotten there yet. So, questions on this? All right, we've done it.
You know what? I did have a question. It might be a little not off topic, but is the myosin always just kind of moving and just waiting for, almost like a transmission, like a clutch kind of thing? It's going until you deactivate or take off the throttle.
Close. And again, we will talk about this when we get to the contractile cycle, but the short answer to your question, it's more like cocking a gun. It is primed and ready to do work, but you can't do work until you pull the trigger, right? Once some event occurs, then it's able to do its work. So yeah, when the muscles relax, those myosin heads are primed and ready to do their work, but they're not doing anything yet because those darn regulatory proteins are in the way. But as soon as they're out of the way, then those primed myosin heads are ready to do their work. All right, excellent. Again, I've done this with my very simplistic drawing. Let's look at the pretty pictures from your textbook. That does a nice job of showing this. Notice here, we very nicely see the anatomy that we were just talking about. Here, of course, that's huge. Here, of course, is the neuromuscular junction, our sarcolemma, that fold of the sarcolemma, the transverse tubules. We see a little bit of the sarcoplasmic reticulum, the enlarged terminal cisternae, and we see how they're wrapped around all of these myofibrils. And then notice again, here we see our single sarcomere of our myofibril. Actually, let's look at this one where we can see it all. Z disc to Z disc, right? We see the actin coming off of that. We see the myosin in the middle. Notice this also has the myomesin down the midline, right? We're not seeing the regulatory proteins at this level of magnification on this illustration, but we can see a lot of that anatomy we have already talked about. Excellent. Here, we see it up close and personal. So again, remember, excitation, contraction, coupling begins where communication at the neuromuscular junction ends, and that is we've produced a muscle action potential and it is traveling down the sarcolemma. And because the sarcolemma folds down into the muscle cell, that muscle action potential spreads down the transverse tubule as well. That transverse tubule is part of our triad. The transverse tubule and the terminal cisternae of one sarcoplasmic reticulum and the terminal cisternae of a second piece of sarcoplasmic reticulum. These two big, huge enlargements of the sarcoplasmic reticulum, which, as we can see, is filled with calcium and has voltage-gated calcium channels. Because that big muscle action potential is a big positive signal that depolarizes the cell. And we forgot to say it, but what's that magical point we have to depolarize the sarcoplasmic reticulum to?
Threshold.
There you go.
60 mil.
Depolarize the threshold, which is negative 60 millivolts. Exactly. Remember, our starting point is negative 70 millivolts, so that's only a 10 millivolt change. And remember, as we've talked about, these action potentials are huge. They're a 100 millivolt wave. So it's very easy for something that is 100 millivolts big to make a small enough depolarization to get it to negative 60 millivolts. And when it gets to negative 60 millivolts, those voltage-gated calcium channels open, and we get an increase in the concentration of calcium in the cytosol. Now let's go back to our picture here for a second. Notice, as we've talked about, when the calcium comes out, these pumps are here. Are they waiting for the muscle to relax to start being active?
No.
No, they're going to be active right away. Some of this calcium is going to, the second it comes out, it's going to get pumped right back inside as well. Right? But this is a jailbreak where we have thousands of criminals leaving the sarcoplasmic reticulum, and this is one poor little Barney Fife. Right? That one little pump's not going to be able to keep up. So there is more coming out than pumped back in. And that is why we get an increase in calcium levels in our cytosol, because more is coming out that can be pumped back in. So the net effect, yes, some of it's getting pumped back in, but a lot more is coming out. So the net effect is we get an increase in the concentration of calcium in the cytosol, not in the cell, because it was all in the cell to begin with. This is just changing location in that cell. Right, from Folsom Jail to Folsom Market, where that calcium can have an effect. Notice here, we are seeing our thread-like tropomyosin, and it is sitting on top of the handrails. Our poor little myosin wants to grab onto that handrail on the actin, but it can't because of that tropomyosin. But when calcium binds to the troponin, troponin undergoes a conformational change. Again, just a fancy way of saying, you don't have to say that on the exam. I like saying it, but you can just simply say it changes shape. And notice when it changes shape, it basically rotates. This one happened to rotate in this direction. So rotated down, the other one rotated around that side. And now our binding sites are open. And now that the binding sites are open, as Brian pointed out, those myosin heads, which are cocked, which are, we can use the term primed, ready. Why is everything on caps? Primed and ready to do work, now can finally do the work that it wanted to do. So our myosin can grab onto the actin, and our contractile cycle is going to begin. All right. Questions on that? All right, excellent. What do we have to do next?
Show how to relax it.
Exactly. All right, we have initiated the process. Now we need to terminate communication at the neuromuscular junction. End it. Excellent. Now, the same way we needed to terminate communication at the neuromuscular junction, where does that begin?
The neuromuscular junction.
True. And what happens at the motor end plate?
Stop receiving NAP.
Excellent. Okay, so if we think back to what happened in the neuromuscular junction. You're right. We stopped producing neural action potentials that repolarized the synaptic end bulb, which closed the calcium channels. Calcium got pumped out. With no calcium, no more acetylcholine was released. But remember, stopping the release of acetylcholine isn't fully what's going to stop the process, because we still have all that acetylcholine in the synaptic cleft. And how did we get rid of that acetylcholine in the synaptic cleft?
Acetylcholinesterase.
We had the enzyme that broke it down.
Diffusion out of the cleft.
Diffusion out of the cleft. And what was the third?
Endocytosis.
Endocytosis. Excellent. We got rid of all of the acetylcholine. Once we got rid of all the acetylcholine, our cation channels closed. With our cation channels closed, no more sodium into the cell. And with no more sodium into the cell, our motor end plate repolarizes and stops producing muscle action potentials. So again, all of that other stuff we talked about was part one. We don't have to worry about that right here. If we're just talking about terminating excitation contraction coupling, this is where we begin. Our motor end plate repolarizes and stops producing muscle action potentials. So that means no new muscle action potentials down the sarcolemma, which of course means no new muscle action potentials down the T-tubule. And if there's no more muscle action potential down the T-tubule, what happens to our triad?
Repolarizes.
Excellent. Triad repolarizes. All right, back to the resting membrane potential. What's, again, the immediate effect of the repolarization?
Voltage gated, calcium channels closed.
Excellent. What's the immediate effect of that?
Decreases the calcium levels in the cytosol.
Well, close. What happens is what closing the calcium channels does is it stops calcium from leaving. So there's no new calcium entering the cytosol. All right? But just like we had before, we have this issue where there's already calcium in the cytosol. So what do we do about that?
Need to get it back into the sarcoplasmic reticulum.
There you go, exactly. We remove it from the cytosol with our calcium pumps. Excellent. Pump it back into the sarcoplasmic reticulum. Again, those pumps were working before, but now that there's no new calcium coming out, these pumps can start having an effect. And we can pump it. And in fact, some of it will actually be pumped out of the cell as well, because we'll have calcium pumps on the plasma membrane, the sarcolemma as well. But the point is we get rid of the calcium. And when we remove the calcium, there's no calcium to bind to the troponin. So what happens to troponin?
Covers back up the actin.
Well, you're right. Well, first, the troponin goes back to its original shape. But you're right. When it goes back to its original shape, it rotates the tropomyosin back over the binding site. This is way too big for this space. Excellent. And when it gets rotated back into the way, I guess we need to move this calcium over. Ooh. Here, bind here to move it. No bind, goes back. And of course, once our tropomyosin is blocking the binding site, myosin cannot bind to the actin, and the contraction stops. And there you go. If you think about it, our goal of termination is the exact opposite of the goal of initiation. We wanted to move the regulatory proteins out of the way. If we want to terminate it, we want to move the regulatory proteins into the way, right? We want to block the myosin. So we wanted to move it out of the way for initiation. For termination, we wanted to move it out of the way. I mean, back in the way. All right, let's look at some more pretty pictures. I like some of these pictures. Again, this one's not from your textbook, from another textbook, pretty simple one. But again, this shows the process of excitation, contraction coupling. We're releasing the acetylcholine, producing an action potential, spreads down the sarcolemma, spreads down the transverse tubule, causes calcium to be released, allowing the myosin and the actin to interact. Although this is actually my favorite picture. Most of the pictures that you see from this, like this illustration here, like this illustration here, show us a longitudinal view. What I love about this one is this one shows it in cross section. Notice what we can see really, really nicely here is, again, here is our binding site on the actin that the myosin heads want to grab onto. But that pesky tropomyosin is in the way. And there's its handle, the troponin. So when calcium is released, it binds to the troponin, causes the troponin to change shape, and causes it to rotate off of the side. Now our binding site is exposed, and now the myosin is able to grab onto the actin, and our contraction can begin. Again, here is a third illustration. Notice at this point it is relaxed. Our sarcoplasmic reticulum is closed. The pumps are moving calcium out. Our voltage gated channels are closed. Our regulatory proteins are out in the way, and our muscle cell is relaxed. But when our action potential spreads down the sarcolemma and down the transverse tubules, it opens the voltage gated calcium channels, releasing calcium. Yeah, we've got those pumps, but more calcium is leaving than can be pumped back in. So our calcium binds to the troponin, moves the tropomyosin out of the way, and myosin can grab onto the actin. And like I mentioned, that calsequestrin is that protein that helps to store massive amounts of calcium inside the sarcoplasmic reticulum. All right. Questions on that?
Professor, could you go back? Oh, there you go. Never mind.
No, perfect. So, any other questions? And remember, I'm saving all these pictures and I'm posting them on our Canvas site so that you have them. They're amazing work of art. You should print them out like posters, put them on the wall.
As long as you can assign the bottom right corner, make it an original.
There you go, exactly. Yeah. All right. Actually, I'm just gonna, like everybody's doing the beer posters now, I'm gonna do a muscle contraction posters. So I'll do that, and they'll be for sale on my my OnlyFans site. All right. Now, if you think about it, as we've talked about with these processes, there's really three things we've been talking about: initiation, termination, And remember, we also talked about modification. Is it possible to modify this process?
Yes.
Yeah, absolutely. It is possible to modify this process. However, modification here is a little bit trickier. Really, the main way we modify this has a lot to do with what we call muscle fatigue. Muscle fatigue is a concept we will talk about more when we build things up. I don't want to talk about it too much here, so it won't be an essay question in relation to this here. So that lops one of our essay questions off, gives us only 5 essay questions so far that we're going to pick 3 of. But just to give you an example of how that may work, as we know, when we use muscle, right, when we use this muscle, 2 things are produced, right? We produce heat, and especially if we are really vigorously using the muscle, we can produce some acids. What acid in particular would be produced if we're overusing the muscle?
Lactic.
Lactic acid, right? And again, we'll talk more about lactic acid later, but the point is we're producing heat and we're producing acids. And what kind of things do we know are really sensitive to changes in temperature and changes in pH? If only we'd learned about
Proteins.
sorry, say again?
Proteins.
Proteins, right? And what is a muscle cell chock filled with?
Protein.
Proteins, exactly. Now, it isn't going to affect all of them equally, but what's interesting, one of the proteins that is most sensitive to pH and temperature changes is our friend troponin here. When the temperature and the pH changes, troponin changes its shape. And now it becomes a lot harder for the calcium to bind to it. And if less calcium can bind to fewer troponins, fewer regulatory proteins are going to move out of the way. And if fewer regulatory proteins move out of the way, what's going to happen to the number of myosins that can grab and pull?
Going to be less. You're not going to be as able, as strong.
And since that pulling of the myosin is what produces our tension, are we going to produce more tension or less tension?
Less.
Less tension. And now your muscle is not working as strong as it was before. Your muscle is fatigued. So just something as simple as that, a little change in the shape of the troponin can affect the efficiency of this process. So like I said, muscle fatigue definitely will be something we talk about, definitely will be something that's a potential essay question, but we're not going to worry about, when we're talking about muscle contraction, we're not going to worry about modification as an essay question there. So again, if you think about it so far, we now have 5 essay questions you're responsible for. Initiation of communication at the neuromuscular junction, termination of communication at the neuromuscular junction, modification. And modification, I won't say how do you modify communication at the neuromuscular junction. I'd probably more do something like how does tetanus, or how does, curare, or if we made this change, how would it affect, right? If we increase the size of the synaptic cleft, what do you think what kind of effect would that have, do you think, on the effectiveness of that muscle contraction? No, I'm asking, if we increase the size of the synaptic cleft, what do you guys think would happen to the effectiveness of that muscle contraction?
I would decrease the ability because more acetylcholine are going to be broken down and not get to the
and it doesn't even have to get broken down, since it's diffusion that brings it to the motor end plate. If you increase the space, more of it will diffuse away, less of it is going to get to the motor end plate, meaning you open fewer channels, meaning you might not be able to get a big enough depolarization to get a muscle action potential. The muscle contraction could actually fail, or at least would be weaker. Absolutely. I actually like that. That's a good essay question. I may ask that. I may add that to the bank. All right, excellent. So those three, and then initiation of excitation, contraction, coupling, and termination of excitation, contraction, coupling. So far, 5 possible essay questions. And like I said, I guarantee you, you'll at least have 2. All righty. Awesome. Questions on this? All right. It is a teeny bit early, but this is a good natural breaking point. So let's go ahead and take our first break here. Or I guess, yeah, this is our first break. First break here. And then when we come back, we will come back and do part 3. So that'll be the contractile cycle. So let's go ahead and take a 15-minute break. Let's say it looks like that we will restart at 9:16. And I will start the recording at that time. All right, see you guys in 15 minutes. All righty. Again, we continue to work our way through a muscle contraction. Someone again remind me what part 1 is?
Initiation?
Well, true. Absolutely. Initiate shall we turn on, but what is the first step we need to turn on?
Oh, communication at neuromuscular junction.
Excellent. And of course, what is our goal here?
Convert NAP into a MAP.
Excellent. Perfect. Step 1. Or first step, let's go with that. Come on, what was the first step? It's the very first thing we have to do if we're going to make a muscle contract.
Decide.
Decide. Excellent. And what was the last step of part 1?
Fluxion potential radiate from the motor plate down the sarcolemma.
Excellent. What's part 2? Come on, we keep beating this dead horse over and over again, so hopefully these answers get easier as we go through them. What is part 2 of a muscle contraction? We just literally finished going over it.
Excitation, contraction.
There you go. Excellent. Goal?
Use calcium to move regulatory protein.
Excellent. What's the first step?
Producing
Muscle.
mechanical action potential at the motor end plate and spreading down the sarcomere.
What was the last step?
Myosin binding to the actin and starting the contraction.
True, but remember the reason myosin was able to grab is because the troponin moves the tropomyosin so that myosin can grab the actin. And since we mentioned it, we should probably throw that in. A primed myosin head can grab the actin. Excellent. That brings us to part 3. And of course, what is part 3 called?
Contractile cycle.
Cycle, or like we also said, the sliding filament theory. Excellent. And our goal here, what's the goal of the contractile cycle?
Contract the muscle.
True, absolutely right. To make the contraction, and again, to really to move it through space. And the way we're going to move the muscle through space is we are going to produce tension. Or again, if we want to think of it in terms of physics, we're going to produce a force. And then that force is going to move our body through space. All right. Again, no new information here, including, what do you think the first step of the contractile cycle is?
Troponin moves to tropomyosin, so primed myosin head can grab actin.
Excellent. There you go. Perfect. All right. So that is where we are at now. Again, I keep beating this dead horse so that we can hopefully make some sense of all of this. Let's go ahead then and start on step 3. Which we know is the contractile cycle. Our goal is to produce tension, force, yammer, yammer, yammer. We know all of that. Excellent. And once again, to fully understand how this works, we need to start with the discussion of the anatomy. Now, a lot of this we already know. This is going to occur in the myofibrils. But remember, those myofibrils are made up of that repetitive pattern of sarcomeres. Let's actually say it that way. And the sarcomeres are the structural units. Again, this is what gives it its stripes. But it is also the functional units which produce the force. All right, so let's talk about the anatomy of our sarcomere. We drew a really simple one earlier, and we can kind of stick with that simple one for starters. We know it is going to be bounded on, why is that green? We have our Z discs, which form the boundaries of our sarcomere. We know connected to those Z discs are going to be our actin filaments. So we'll put an actin filament there, and an actin filament here, and another actin filament here, and another actin filament there. We know between them, we have our myosin filament, that thick filament. And we know that the myosin filament has those myosin heads. Now, we know myosin heads can be primed. The implication there is that they can also be not primed. And one of the things that we haven't talked about yet, but is probably worthwhile to talk about now, is that a primed myosin head points away from the M line. Now, of course, we haven't drawn the M line yet, so let's go ahead and draw our M line. Our M line, as we know, is that myomesin, those 3 rows of interlocking proteins that help to stabilize not just this myosin in place, but remember down here, there's another myosin, and there is another myosin, and this is a 3-dimensional structure. And these actually also cross the myofibril to the myofibril next to them to hold those in place as well. So notice our myosin heads here on the right are pointed away from the M line when they are primed. And obviously these aren't drawn to scale, but I need them big for the next thing we're going to talk about. Pointed away from the M line. So the ones on the right point to the right, the ones on the left point to the left. So you could also see how they're pointed towards the Z disc. I guess you could say it either way. They're pointed towards the M line or they're pointing towards the Z disc when they are primed. So I guess then the next question becomes, what does it mean for a myosin head to be primed? Well, remember, a myosin head is a motor. Someone remind me again what a motor does? Anyone remember?
Creates mechanical power.
From what?
Energy.
Right, exactly. I like that. That's good. Absolutely. Its job is to convert energy. In this case, it converts a chemical energy into a mechanical energy. And of course, what is the chemical energy it uses?
ATP.
Yeah, ATP, absolutely. Remember, one of the things we talked about with our ATP is that ATP is that adenosine nitrogen base connected to a ribose sugar with not one, not two, but three phosphates bound to it. So when we split ATP, we basically rip one of the phosphates off. And as a result of that, our ATP releases energy, or as a result of that ATP getting ripped off, or split, we get a tremendous amount of energy from breaking that bond, plus we get obviously the phosphate that's gotten ripped off, plus what are we left with?
ADP.
ADP, adenosine diphosphate, as a result of it. And so that's what a myosin head does. The myosin head splits, well, let's say it this way. To prime a myosin head, it splits the ATP into ADP and phosphate. But it doesn't use the energy right away. It stores the energy. So in this primed myosin head, there is an ADP, there is a phosphate, and there is energy that it is just waiting to use. But it can't use the energy until it binds to the actin. So primed, it really is cocked like that gun, ready to go off, ready to do work. Has that energy and it's ready to use it, but it can't use it yet until it comes in contact with that actin.
Is that just like potential energy?
Yeah, essentially it's a stored potential energy. It's a stored kinetic energy, right? Just like I have that ball that I'm holding at the top of a hill, it has a tremendous potential energy. And as soon as I let go, it rolls down the hill, right? As soon as the regulatory protein is removed, it's able to bind. And when it binds, it's finally able to release its energy. Okay, since we've done it, let's go ahead and add a couple more, not as relevant to the process, but don't forget that we also have that titin protein. That is what stabilizes the myosin. Again, it doesn't play a huge role in this process, but we'll put it here just for the heck of it. But what we also need to remember is, again, we have that regulatory protein, the tropomyosin, which is blocking the binding site, and its handle, the troponin. All right. Questions on this? Notice we've done a good job of drawing this here. If we go back to our lecture slides, here we see another simple illustration of a sarcomere at rest. Again, it goes from Z disc to Z disc. Z discs have the actin bound to it. Myosin is held in the middle by the myomesin and the titin. Notice the titins here are just connected to the end. We know that's not the case, that it actually glides across the side and actually anchors to the myomesin, but that's okay, it's simple enough. And the other nice thing about it is it reminds us of the stripes. As we mentioned, the sarcomere, its organization give us the stripes. We have the dark band, which contains the myosin, the I band, which doesn't contain the myosin. The center of the I band is a Z disc, because I band bridges to sarcomeres, whereas an A band is completely contained within a sarcomere. In that A band, there's a lot of myosin and actin overlapping, but there's also a region that just has myosin in it. And that area that just has myosin in it is the H band. And of course, the myomesin interlocks with the myosin at the center, and that of course forms the M line. Again, no new information here. We've understood this, we've talked about this. This should hopefully be making sense at this point in time. And this is our resting sarcomere. And so what our book has done and what we are going to do is we are going to take a small portion of a sarcomere from over here on the right-hand side and look at it up close. And notice that's what they have here. This picture from your textbook is excellent. We have our dimer actins. We see the yellow that represents the binding site, our thread-like tropomyosin, the handle-like troponin, blocking those binding sites. And notice, because we're here on the right, we have some primed myosin heads. They've split the ATP into ADP and phosphate. Notice your book doesn't emphasize that they're still storing the energy, but they are. And notice those primed myosin heads are pointing away from the M line because we're over here on the right. So this is our starting point. All right.
Isn't that picture of the sarcomere? That's not a resting sarcomere.
Well, yeah, notice that our artists, the blue lines that have represented the heads, they do have pointing in the wrong direction. Yes. A little bit of artistic license they're taking on this part right here. Absolutely. Although it looks like they've at least got the arrows. So it looks like maybe it's contracting. So maybe that's what they're trying to show. However, I will tell you, do you think that all the myosin heads all pull at exactly the same time like this one is? No. So again, a little bit of artistic license. But I want us to focus on here, what's going on here in this resting sarcomere. All right. Are we comfortable with the anatomy? Because again, if you don't understand the anatomy, it only gets worse from here. All right, excellent. Then here's what we're going to do. We are going to go back to our whiteboard and we'll go through it together here. I don't remember if I saved the anatomy of this, so I'll go ahead and save this picture again. Excellent. And now go ahead and clear that. We are going to now do part 3. Contractile cycle. And again, I'm going to leave it cycle because remember, cycle means it's a continuous process, and that is going to help us. And of course, the goal, as we talked about, is to generate a tension Or again, if we're thinking in terms of physics, we want to generate a force. All right, let's do some anatomy. We know we have our actin connected to the Z disc. We know we have our myosin, which we'll put here, portion of it anyway. And here we have the myosin head. Notice I have my myosin head here. Actually, that's huge. Pointing away from the M line. So of course, this means it must be a primed myosin head. Which means that it's already split an ATP into ADP, into phosphate, and into energy. Ready to use that energy, ready to do its work. And of course, as we've talked about, this process has to begin with moving the regulatory proteins out of the way. So there's my troponin, here's my tropomyosin. We'll draw an arrow to remind ourselves that this has been moved out of the way. And of course, it's been moved out of the way because calcium has bound to it. So again, our goal here is to understand the process of initiation of the contractile cycle. And this is our starting point. With our starting point, we have the regulatory proteins are moved out of the way. And when that tropomyosin is moved, then our primed myosin head is able to grab onto the actin. And so that is what's going to happen. So let's go ahead and continue our drawing over here. There's my actin. There's my Z disc. Here's my myosin. And here is my myosin head. With the ADP, the phosphate, and the energy. So with the regulatory proteins out of the way, myosin is finally able to bind to the actin. Which is fun and easy and simple to say. But of course, as we know, anatomists hate us, so they have to give a fancy name for this. This binding of myosin to the actin is what we call the crossbridge. So basically, our myosin head forms a crossbridge. But again, forming that crossbridge just means that it grabs onto the actin. With those regulatory proteins, and again, we should emphasize it, our primed myosin head binds to the actin, forming that crossbridge. And once it forms that cross bridge, our myosin head is finally going to be able to release the energy. It's finally able to do its work. And in this case, its work is going to be, again, another fancy term, perform a power stroke. So what the heck is a power stroke? Well, basically what it does is our myosin head pivots towards the M line. So again, let's work on our drawing. I don't like my wiggly actin. So there's my actin, my wiggly Z disc, my myosin, and what my myosin head has done is performed this power stroke. Basically, it has pivoted. So again, it started here. Oops, nope. It started here, and it pivots towards the M line. Now notice, as it pivots towards the M line, it is going to pull on the actin. And guess what? Our actin is connected to the Z disc. So it pulls the Z disc towards the M line. Now remember, this is happening on both sides. On the right, it's grabbing and pulling towards the middle. And on the left, it's grabbing and pulling towards the middle. So if you think about this, as the myosin heads on both sides perform power strokes, both Z discs are pulled towards the middle. So what happens to my sarcomere?
Decreases in length.
Yeah. My sarcomere gets shorter. And obviously this is happening in all the sarcomeres, but even if it was just happening in this one, if my sarcomere gets shorter, what happens to my myofibril?
And
It's
also,
short.
And of course, my myofibril with those structural proteins is connected to the sarcolemma of the cell. So if my myofibril gets shorter, what happens to my muscle cell?
Get shorter.
That muscle cell is surrounded by connective tissues, right? The endomysium, it's in a fascicle that is a perimysium connected to the epimysium on the outside. So as my muscle cell gets shorter, my entire muscle gets shorter. And this, we accomplished our goal. We're changing the shape. We're generating our force. We're generating our tension. Mission accomplished. Just that simply, just that easily, we have generated some force. So again, this myosin head uses the energy and performs the power stroke. Oops, that doesn't need to be caps. And that power stroke generates our tension. All right. Questions on that? Now, we've generated our tension. Does that mean we're done?
No.
No, of course not. This is a cycle. And one myosin head performing one power stroke isn't going to be enough to move my finger entirely through space. So some other things need to happen as well. But this right here, this power stroke, is where we're performing our force, where we're generating that tension. But we want to keep doing it. After all, this is a cycle. Now, one of the ways that allows this to happen is there's one other event that occurs during this power stroke. During the power stroke, the myosin head releases the ADP and the phosphate. After all, it's used the energy from them. It's been able to release that energy. It doesn't need them anymore. So during the power stroke, the myosin releases the ADP and phosphate. So here's the ADP. Here's the phosphate being released during that power stroke. That's useful because remember, here inside of our muscle cell, inside of our myosin head, our binding site for ATP, the ATP that came in there before and was holding before, is now open. And if you have an open binding site for ATP inside of this myosin head, what might conveniently bind there for us? It's not a trick question.
Some more ATP.
Yeah, exactly. Absolutely. And that's exactly what happens. Notice at this point, our myosin head has done all of its work. It's used all of its energy. It's released the ATP. It's done all the things that it needs to do. But it's going to want to do it again. So what we're going to need is to, let's do it this way. A new ATP binds to the myosin head. When this occurs, our myosin head undergoes a conformational change. And as a result of that, the myosin head releases the actin. After all, as we've talked about, I'm climbing a ladder. I grab the ladder, I pull myself up. If I want to keep that process going, the next thing I need to do is let go of the rung. And that's exactly what happens. Binding a new ATP to the myosin causes the myosin to let go of the actin. Basically, what we say is it breaks the crossbridge. So our myosin head, when a new ATP comes and binds to it, that myosin head lets go. Just like my hand. I grab, I pull, I let go. We break the crossbridge. Notice it's not using the energy from the ATP. This is just a conformational change. Just letting go. Of course, now that I've let go, am I ready to do work again? Am I ready to pull on the rung of the ladder again?
No.
No, I need to reach out so that I'm ready to grab and pull again. Notice I've bound this ATP, I've broken the cross bridge, but the myosin head is not primed. And if it's not primed, it's not ready to do work. And how do we get this myosin head ready to do work?
Leave one of the phosphates.
There you go. Our myosin head then splits the ATP into a DP and phosphate and energy. When it splits the myosin head, this primes the myosin head, and the myosin head points away from the M line. And of course, and here's the key word, if the regulatory proteins are out of the way, the process continues. The process or cycle continues. There you go. There is our contractile cycle. It really is like climbing a ladder. Prime, grab, pull, let go. Reach, grab, pull, let go. Reach, grab, pull, let go. Reach, grab, pull, let go. All right? Myosin head primes, forms a cross bridge, power strokes, breaks the cross bridge. Primes, form cross bridge, power stroke, break cross bridge. Prime, and around it goes.
Professor, could you move up the word groups on the bottom right and the bottom left, please?
Yeah, let me see what I can do with that. If I move that up there, is that high enough there?
Yes.
And then there.
Perfect.
Excellent. And there you go. That is our contractile cycle. Questions on this? All right, excellent. Let's look at the pretty pictures from your textbook. Again, we start with a resting sarcomere. I like this picture from your textbook a lot, although you'll see there's one part I don't like. Again, in that resting sarcomere, when the muscle is relaxed, the troponin and tropomyosin are in the way. Our myosin heads are primed. Even though your body is at rest, that cell has plenty of ATP in it, and so any myosin head's going to grab any random ATP around, split it, prime it, hold on to the energy, and be ready to do work. And of course, it's storing that energy, and it is pointed away from the M line. That is our starting point. That is our anatomy. And our muscle is relaxed. But when we move those regulatory proteins out of the way, Calcium binds to troponin. Troponin rotates to the tropomyosin. And now our active site is revealed. This, of course, was the end of excitation contraction coupling, and therefore it's the beginning of the contractile cycle. Once those binding sites are exposed, what's the first thing that's going to happen?
Formation of the crossbridge.
Exactly. Our myosin head is going to grab onto the actin, forming that crossbridge. That energized or primed, cocked, whatever term you like, right? Whatever term you like, which basically means ready to do work. Right? So that energized myosin head, that primed myosin head is going to form a crossbridge, binding to the actin. Right? Remember, only the primed myosin heads are able to do this. They've already split the ATP, storing the energy, and they're pointed away from the M line. Once they form the crossbridge, they perform their power strokes. Right? It's a fancy term, but all it means is that myosin head pivots, rotating towards the M line. It releases the energy while it's doing this. That pulls the Z disc towards the M line, pulls the actin towards the M line, makes our sarcomere shorter, makes our myofibril shorter, makes our muscle cell shorter, makes our muscle shorter, generates our tension, right? This power stroke is where we generate our force. This is where we're generating our tension. And remember to keep the process going during this power stroke. The myosin head is also letting go of the ADP and phosphate. This is the picture I don't like. Once our power strokes, let's go back. Once our power stroke is completed, now a new ATP can bind to the myosin head. And when it binds to the myosin head lets go. Right? So our next step is letting go, and that is the breaking of the crossbridge. And again, remember, this is a conformational change. It's not using the energy from the ATP to break the crossbridge. It is just letting go. Notice here, your book has already primed the myosin heads and split the ATP, so they skipped a step here. But I have a better picture that I like that I'll show you in just a minute. So again, binding of the ATP changes the shape of the myosin head and causes the myosin head to let go of the actin. Now, the hand hole on the actin is open, so another myosin head could grab on it. But this myosin head's got to revitalize. It's got to split that ATP, forming the ADP, forming the phosphate, and that reprimes, re-energizes, recocks the head so that a new crossbridge can form and the process can continue. Here's the picture I like a lot. This one's not from your textbook, but I really like this picture. I think it does an excellent job of showing the process. Notice here we start with a primed myosin head. It's got its ADP and phosphate. Our regulatory protein's out of the way so it can bind, forming that crossbridge. Once it forms the crossbridge, it is going to perform its power stroke, pivoting towards the M line, pulling on the actin, pulling on the Z disc, shortening our sarcomere, shortening our myofibril, generating the force, and releasing our ADP and phosphate so that a new ATP can bind to it. Notice just the binding of the ATP breaks the crossbridge. The myosin head changes its shape and it lets go. but that myosin head's not ready to do work until it splits that ATP. When it splits the ATP, that primes the myosin head. It has its energy. It's ready to work, and this process continues. Again, reach, grab, pull, let go. Reach, grab, pull, let go. Each contractile cycle takes a fraction of a second. An average myosin head can perform 5 contractile cycles in a second, right? 1, 2, 3, 4, 5 of those, all within 1 second. And every single myosin head grabbing and pulling shortens the muscle cell about 0.5%. And as we've talked about, if it's just me with my 2 hands and I'm climbing the ladders, do I reach with both hands at the same time, grab with both hands at the same time, pull with both hands at the same time, and then let go with both hands at the same time?
No.
No, I'm going to be alternating what these are doing. And that's just with 2 hands. Our myosins have thousands of heads. Those thousands of heads are all going to be grabbing and pulling at different times. So while one's grabbing, one's letting go, while one's reaching, one's revitalizing, and they're just going. And how long will this process continue for? How long will this contractile cycle continue for?
Until you decide not to flex.
True, but what is it about the decision? Tell me literally, what keeps this contractile cycle going?
The calcium
Anything else?
on the calcium attaching to the troponin, which allows the conformation of the tryptomyosin.
Okay. Is there anything else that's needed to keep this process going?
ATP.
ATP. As long as there's calcium and ATP available, this process will continue. Which, if you think about it, is how we turn it off. As we talked about, we pump the calcium back into the sarcoplasmic reticulum, and with no calcium, no regulatory proteins move. And when no regulatory proteins move, suddenly that primed myosin head cannot grab the actin. And if it can't grab the actin, no more tension. Bingo. I guess for consistency, we should make this red, because essentially what we just talked about is termination. However, if you notice, the termination process is really simple. So simple, is it the kind of thing I'm likely to give you as an essay question?
Yes, because you like us.
No. Right? But we understand it. That's how the muscle relaxes. We get rid of the calcium, the regulatory proteins move back in the way, and that disrupts the cycle. Now, technically, couldn't the cycle also stop if we run out of ATP?
Yeah.
Technically, that is the case, but do our cells ever really run out of ATP?
No.
Never?
Most of the time they don't.
Most. When might be a case where your cell would run out of ATP? I know you know the answer to this, even if you don't know you know the answer to this. Let's think outside the box. when would your cells not be making or producing ATP? When would your cells no longer have ATP production going on? Come on, I know one of you knows it.
Well, I'm thinking like the citric acid cycle, but that's probably too far
Arabella's
back.
got it. Arabella hit it on the head. Death. When you die, are your cells producing ATP anymore?
No.
No. And you've probably never thought of it in these terms before. But if you've ever seen like an NCIS or a CSI or any of those types of shows, right? One of the things that happens when they find a dead body, depending on when they find the dead body, is when the dead body first is found, there you go, Jacob's got it. When the body's first down, it's all, limp wherever it is. But if the body's been sitting there for a while, one of the things that happens is rigor mortis. What is rigor mortis?
Contracting of your muscles.
Yeah, well, basically what happens is the body becomes locked rigid into whatever position it was in when it died. Right? Exactly. And that's what happens, Jacob. So let's think about this. You die and you stop making ATP. Your myosin heads have already grabbed lots of ATP. So as a result of that, they're already primed. Oops. We've got primed myosin heads. But without ATP, things like your calcium pumps can't be working anymore. So as the ATP in the cell is used up, and the cell, because it's dead, is not making anymore, calcium is going to start sneaking into the muscle cells. And when calcium binds to the troponin, it's going to move that tropomyosin out of the way. And that primed myosin head is going to grab and pull, performing its power stroke. But there's no ATP left in the cell. So can the myosin head let go?
No.
No. And so, no, Botox remembers something different. We'll go back to that one in a second. So now the myosin heads can't let go. And so basically what happens is the muscle cell becomes locked into that position they were in when they died. Now, does rigor mortis last forever?
No.
No, because as the body starts to deteriorate, the actin starts to break, the myosin heads break off, the Z discs start to break. As the proteins start to break down, then the structure is lost, and then the body becomes flaccid again. Right? We can actually get a sense of how long someone has been dead by where they are in that process. Right? They have all these tests, temperature, all these other types of factors that they can take into it that help them to know how long will it take for rigor mortis to set in, how long will it take for the rigor mortis to go away, and they're able to, by that, determine how long someone has been dead for. It's one of the main factors they use in determining how long someone has been dead for. Excellent. Now, remember, with Botox, what it does is the proteins that are breaking are actually in the synaptic end bulb. So it's not actually damaging the muscle cell. If you were to artificially dump acetylcholine onto a muscle cell, that muscle cell would contract. Or if you stimulated it with an electrode, that muscle cell would contract with Botox because it's just causing no acetylcholine to be released. So that is also a flaccid paralysis, but that in that case is from damaging the neuron, not from damaging the muscle.
So I totally had a question like popping in my head earlier. It's way off topic, but when you get like a muscle twitch and everything, like
you got the right idea. So notice we have now seen how a muscle can contract. So typically when someone has like a Charlie horse, a muscle cramp, or a twitching, one of two things are happening. We know we voluntarily control our muscles, but now we know we do that by releasing acetylcholine that allows sodium into the cell. So basically we have neurotransmitters that open channels and ions that move into the muscle cell. So the two most common causes of twitching or cramping is either if your nerve is irritated or something like that, where it is inappropriately releasing neurotransmitter, that could cause the muscle to contract inappropriately. And then often an ionic imbalance. If the ion imbalance is off, if the ionic balance is off, that can change the flow of ions into or out of the cell at rest. And as you've seen, we only have to get from -70 to -60. So it doesn't take a huge imbalance of ions to get it to change its membrane potential to a point where it could cause the muscle to twitch. So there are other factors that can cause it, but those are the two most common. Irritation to the nerve where the nerve is firing inappropriately, or ionic imbalance triggering a muscle action potential when you don't want one in the muscle cell. Awesome. Excellent. How are we doing? We're doing good on time. All right. It's a little long, but I want to take a second to do this. All right. Any questions on this before we move forward? All right, excellent. So if you think about it, I have told you numerous times now about having at least 2 possible essay questions, guaranteeing you that you are going to have 2 essay questions on this muscle contraction process. And now, mostly, we have identified what all of them could possibly be. So let's go through them together. Possible essay questions. Give me one of the possible essay questions that I could give you on this exam.
Describing the process of initiation at the neuromuscular junction.
Excellent. So excellent. So initiation of communication at the neuromuscular junction. What's the second one?
Termination of communication at the neuromuscular junction.
What's another one?
Initiation of muscle action potential at the motor end plate.
So I see where you're trying to go with that, but remember, how am I going to ask the essay question?
Excitation and contraction couple.
Wait, why did I number that 4? Did we miss one?
I think Jacob got it. Modification of neuromuscular junction.
However, again, let's write this in terms of how I would do it on the exam. Yes, absolutely, modification of communication at the neuromuscular junction is one of the questions I'm going to ask, but that's not how I'll ask it. How I'll ask it is how would X modify communication at the neuromuscular junction, and what effect would it have on the muscle? So for instance, X could be tetanus. And as we know, tetanus binds to and keeps the cation channel open, allowing sodium to continuously to flow in. And the effect that has on the muscle is a rigid paralysis. So you're right. Question 3 is modification at the communication at the neuromuscular junction. But unlike initiation, where I'm going to ask, describe the process of initiation of communication at the neuromuscular junction, I would ask you more specifically about one of the toxins, or come up with something else like we talked about, like increasing the space of the synaptic cleft or something like that, where you show your understanding of this process by understanding how that would change the impact on the muscle. Excellent. What's next?
termination of the excitation and contraction coupling.
And
Initiation of the contractile cycle.
And honestly, since it's just initiation we have to worry about, I could just simply ask you to describe the process of the contractile cycle, because we're only really responsible for initiation. So there you go. Those are the 7 possible essay questions that I guarantee you will have 2 of them that you will be responsible for on the exam. All right. Questions on those 7 essay questions? No one has any questions about those 7 essay questions?
Well, what's the 7th?
Yeah, there you go. I was wondering for that. Absolutely. There is indeed a 7th possible essay question. And I know you're freaking out right now. Oh my God, he's going to teach us even more stuff. No, there is absolutely positively no new stuff on this last question. But remember, at the very beginning, we talked about how potentially there could be a 4th step in this process. And what would the 4th step in this process be?
The muscle relaxes.
Yeah. But the good news is there's no new information there. If you think about it, how a muscle relaxes is really just termination of communication at the neuromuscular junction and termination of excitation contraction coupling. So let's do that. Let's put them together, understanding how we need to answer our essay questions by making sure we emphasize every initial step in the process. If one of your 2 essay questions, and I guarantee you there's a test bank that has these 7 questions in it, and you're getting 2 of them. Guaranteed. Next exam, that's what it's going to be. And are there plenty of other questions that are like multiple choice questions and other things that are going to involve these questions? Absolutely. So let's do this. Let's put it together. If you got the question, how a muscle relaxes, how would you answer that question? Let's put the pieces together. How do you get a muscle to relax? What's step 1?
Decision in the brain to not.
Excellent. And when you make the decision to relax the muscle, what happens?
No new neural.
Excellent. Stop making neural action potentials. No neural action potentials. There's no new big positive. Oops, so I suppose positive signals spreading down the axon to the synaptic end bulb. And if there's no more neural action potential spreading to the synaptic end bulb, what happens to our synaptic end bulb?
Repolarizes.
Excellent. What's the immediate effect of the synaptic end bulb repolarizing?
Voltage gated calcium channels close.
Perfect. What's the immediate effect of that?
No acetylcholine.
No, it's not the immediate effect. When the channel's closed, there's no
No new calcium into the synaptic end bulb.
But we still have a problem. There's still a lot of calcium in the synaptic end bulb. So what do we have to do?
Pump it out.
Excellent. And now you're right. With no calcium in the synaptic end bulb, what happens?
No more exocytosis of acetylcholine.
Excellent. No new acetylcholine is being exocytosed from the synaptic end bulb. Solves half of our problem, but there's still a lot of acetylcholine in the synaptic cleft that we need to get rid of. And how do we do that again?
Acetylcholine esterase.
Excellent.
Endocytosis.
Excellent.
Diffusion of the acetylcholine out of the cleft.
Excellent. Again, remember, acetylcholine esterase is the primary way, but we still have endocytosis, we still have diffusion. So even if acetylcholine esterase went away, eventually we'd be able to get rid of the acetylcholine. It would take a lot longer, but eventually we'd still be able to get rid of it. Excellent. So now we have no acetylcholine in the synaptic cleft, and what is the effect of that?
Chemically gated cation channels close.
Excellent. Why is that important?
Doesn't allow any new sodium into the muscle fiber.
Into the motor end plate. Excellent. Yes, no potassium leaves either, but remember, it's the sodium rushing in that is causing the effect. So really, we don't care about the potassium so much in this case. It's really the sodium coming in that we care about. I put it there just for consistency, but again, it's not important for the relaxation process. Excellent. With no new sodium coming in, what happens?
The cell repolarizes.
Yeah, our motor end plate goes back to the resting membrane potential. Or I like the way you said it, repolarizes. Excellent. So what happens when the motor end plate repolarizes?
No new muscular action potential.
Excellent. No new muscle action potentials. All right, so also no muscle action potential spreading down the sarcolemma and down the transverse tubule. Excellent. What's the effect of no muscle action potential down the transverse tubule?
The triad gets repolarized.
And why is that significant? Although we should probably say what the triad is. What is the triad?
Transverse tubule. And the Sarcoplasmic Reticulum.
Yep, and specifically the terminal cisternae. Excellent. So our triad repolarizes. And what happens when our triad repolarizes?
The voltage gated calcium channels close.
Excellent. So of course the effect of that is no new calcium into the cytosol. But again, it's only half of our problem. We still need to get rid of the calcium that's already in the cytosol. And how do we do that? Calcium pumps, excellent. And with no calcium, what happens next?
The tropomyosin goes back.
Why does it go back?
Because calcium isn't binding to the troponin.
Excellent. So without calcium, no calcium in the cytosol, troponin goes back to its original shape. And you are correct. That rotates the tropomyosin back over the myosin site on the actin. Myosin cannot grab the actin, or heck, we can be more specific now. Form a crossbridge with actin, and with no crossbridge, muscle relaxes. I appreciate this is a little intimidating to look at, but there really is no new information here. All right. If you think about it, this is just essay question 2 and essay question 5 together. In fact, we can actually divide this into the 2 steps. Obviously, question 2 begins with making decision. Where would, if you got essay question 2, this is exactly what you would write up to what point? Where would essay question 2 stop?
No.
No new sodium into the motor input.
Nope.
No new muscle action potentials.
There you go.
Potentials.
All right, that right there would be essay 2. And also remember, that would also be essay question 5. So notice if we break it this way, this right here is essay question 2. And this right here is essay question 5. Oops. So really, 7 isn't a new essay question. It's just putting the pieces together. So it really isn't that scary. It shouldn't be. You should know exactly what to expect from this. Again, I can't emphasize it enough. On this next exam, it might even be the first essay question, first couple essay questions, but I guarantee you there is a test bank that has these 7, really a little bit more because different modification types in there. But basically these 7 essay questions in it, and you're getting 2 of them. 100% guaranteed. All right. So hopefully this does 2 things. Hopefully this helps you to both prepare to know what to expect on this exam. But remember, the other thing that I've been trying to emphasize is to give me the details. An easy way to do that is to make sure that you think about what's the immediate effect. Many of you are writing essay questions that are 100% correct, but they're lacking detail. Right? I could answer the question, how does a muscle relax, by saying, I make the decision to relax the muscle, I stop releasing acetylcholine from my neuron, my muscle cell repolarizes, and the myosin and the actin don't interact anymore. And that essay question is 100% correct. But I'm lacking all of the specifics. I'm lacking the details. And so while that essay question would be 100% correct, of the 8, 10 points that you would get for that essay question, you might get 2. Because you're lacking all the detail. The detail is how you show me that you understand these processes. All right. Questions on that? All right, that went a little long, but that's okay. I wanted to make sure, I think this is important to go over this. I want you guys to be successful. I want you to understand what is expected of you. And again, you've already taken 2 exams. You've seen that the way I ask my essay questions, the way I ask my lab questions doesn't change. You now know what to expect from me on the exams. And so if you're not being successful, I'm not going to change. You're going to need to change the way you're attacking this material, or more importantly, answering these questions. One of the challenges to a class like this is there's really 2 skills you have to learn. The first is how to cram a whole bunch of information into your brain. But the second thing you have to learn is how to present that information on the exam. And that's the part that's more specific to the instructor, right? The femur is the femur is the femur. It doesn't matter what anatomy and physiology instructor on the planet you speak with, the anatomy of the femur is not going to change. But how they ask their essay questions and how they grade them, that's where instructors are different. So I'm trying to do everything I can to show you exactly what it is that I want that will help you to be successful in my class. And if you're successful in my class, I guarantee you you'll be successful in anybody else's class. So whether you take me for 431 or somebody else. And more importantly, when you take your CAP tests and all those other tests, this is the stuff that you're going to need to know. So learning it now is going to help you to be successful on that. All right, off my soapbox. I'll put my pom-poms away. Let's go ahead and take our last break, and then we'll come back and switch gears into the origins and the insertions. It is 10:33, so we will restart at 10:48. Grab your red and blue pens, and I will meet you back here in 15 minutes. Any questions? All right, see you guys in 15 minutes. All righty. Let's go ahead and get started. We left off last time and we had talked about the muscles that move the head. So now we are going to focus on muscles that move the trunk or the core of the body. And we're going to start first with the trapezius. Notice, as you can see, the trapezius is a big, large, convergent muscle. So we know that's going to provide for lots of flexibility of movement. And if we were to describe it, notice it is superior, it is superficial, and it is mostly posterior. Obviously, this is a big prominent muscle on the posterior part of the body, but notice that pesky word mostly, because what, of course, does mostly mean?
Not all.
Not all the time.
Not all, absolutely. And we see that a little bit with the fascicles, if we look at the way the fascicles go. But I think where we see this best is when we actually draw the origin and the insertion. So, of course, let's go to our drawing for the origin and insertion. And it looks like my trapezius is on page 2. There we go, perfect. And make this a little bigger. I think we can still fit it in there. Excellent. Oops, no. All right, excellent. So let's start first, as we've been doing, with the origin. What is the origin of the trapezius muscle?
Occipital bone, ligamentum nuchae, and spines of C7 in all thoracic vertebrae.
Excellent. So when we're talking about the bony features, it's that occipital bone, again, that external occipital protuberance that we've talked about, the spinous process of C7, that vertebral prominence we've seen, and then 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. Excellent. The spinous processes of all the thoracic vertebrae. Now notice in there also was the ligamentum noche. Ligamentum noche, remember, is that elastic ligament made up of elastic connective tissue that helps to hold our head up. And so notice if the trapezius muscle doesn't directly connect to the cervical bones except for C7. But if you think about it, makes sense. Our cervical bones, we need that flexibility of movement. So it kind of makes sense that we wouldn't want to anchor it, a big, huge muscle like this, to the vertebrae of the neck, because that might restrict their movement. So notice this gives us essentially a continuous median insertion, just not all of it is two bones. All right. Excellent. So now let's talk insertion. What is the insertion of the trapezius?
Continuous insertion along acromion, spine of scapula, and lateral third of clavicle.
Excellent. So along the spine, out to the acromion, which of course, as we know, are posterior bone features. But notice also, it actually comes a little bit onto the clavicle, the lateral third of the clavicle. So some of the fascicles actually do insert onto the anterior side of the body, over here onto the front of our clavicle. This gives us this very, like I said, convergent looking muscle. It's where it gets its name, basically, because we have all these fascicles coming in here and here like this. So basically it's a trapezoid shape, two trapezoids on both sides. You have this big kite-like superficial superior muscle on the back side. And again, this convergent muscle that attaches to the head and the vertebral column to give us that flexibility of movement. As we mentioned, our pictures do a decent job of showing this. Notice now that we know what we're looking for, we can see how some of the fascicles from the superior part of our trapezius disappear because they're going on to the front of the anterior part of the body. But it might not be as noticeable when you're feeling it on yourself, unless you happen to look like this fella here. If you happen to look like this fellow here, notice what we see here is this big, huge bump that we see on his neck, right? This is those fascicles from the trapezius coming down onto the clavicle. Especially when he's leaning forward, you can see how pronounced they can get. So here again, with our bodybuilder, it does a really nice job of showing us the uniqueness of this insertion for the trapezius. How again, it is mostly on the back, but there is a little bit of it that creeps onto the front as well. All right, questions on that? All right, now not surprisingly, with a convergent muscle, there's going to be a lot of actions. And in fact, how many actions does the trapezius have?
4.
4. What are they?
Raise, elevate, rotate, and retract the scapula and extend the head.
Excellent. Absolutely. So you went through those fast. So let's quickly go through them together a little slower. Well, quickly slower, that doesn't make any sense. But let's go through them again. Absolutely. Elevate the head, or pardon me, extend the head. Extend the head. So again, increases the angle. So it brings the head back, looking up into the sky. And then it has 3 effects on the scapula. They were basically on the pectoral girdle that it attaches to. Elevates, retracts, and also rotates. And again, a good way to remember this are the 3 R's. Way back in ancient times, and by I mean ancient times, I mean when I was elementary school, what were the 3 R's that I learned in elementary school? Anyone know?
Recycle, reduce, reuse.
What? Say that again.
Recycle, reduce, reuse.
Okay, I appreciate that. I'm not nearly that young. That was the one that my daughters learned. My daughters learned reduce, reuse, and recycle. That was their 3 R's, but I'm much, much older than them. And in ancient times, when I was in elementary school, the 3 R's we learned were reading, writing, and arithmetic. And if you think about it, 2 of those 3 things aren't really R's, but that tells you something about the education we got back there. However, here we have the 3 R's of the trapezius, right? R1 is to elevate or raise the scapula, brings the scapula up. R2 is retract. Remember, retract means to bring back. Now, remember also, as we talked about, when you're bringing the scapula back, you are also pulling it towards the midline. So we could also use a deduct for that as well. And rotate. Rotate gets a little bit trickier. This is something you can kind of feel for yourself, but it's actually easier to feel on somebody else. So if you have someone else living in the house with you, then I encourage you to have them do this. If not, next time you're at the supermarket, just randomly put your hand on the back of somebody. And when you put your hand on the shoulder place of just some random person in the store, ask them to then abduct their arm. And as they abduct your arm, what you'll notice as you are moving your humerus from your side up to about here, you're just moving the arm. But remember, we have a bursa here and we have an acromion here, and we have all these things that could bump into the neck of the humerus. So as you continue to abduct the arm, what actually happens is your scapula has to rotate, so that the humerus and the scapula basically rotate as you continue to do that abduction for your jumping jack. So you can kind of feel it on your own scapula, but like I said, it's much easier to feel it on somebody else's. So those are the 3 actions. We are able to raise it and bring it up, retract it and bring it back, and then also rotate it. So 3 R's for the trapezius, for the trapezius on the scapula, and extend the head. Now, hopefully you guys have been working on your activities. Does anybody have a good activity that involves all of this for the trapezius? Helping us to remember the trapezius and all these actions?
Throwing a ball up for a tennis serve? Would that be the one?
Absolutely. That's actually my favorite. Absolutely. I love that, right? Because again, if you think about you're getting ready to do a tennis serve, what you are going to do is you're going to bring your shoulder up, bring your shoulder back. You got to rotate it up so that you have the handle of the tennis racket there ready to go. And when you throw the ball up in the air, you have to look up to see it so that you're ready to hit it. Absolutely. That's a perfect one. Excellent, excellent. So great example of, again, coming up with a common activity, or not common, I don't know how many people play tennis anymore, but common activity, or at least an activity that we can use to help us to remember all of the actions of the trapezius. Perfect. Questions on that? All right. Let's talk about another really interesting muscle here, the serratus anterior. I like these pictures from your textbook. I think they do a decent job of showing them. Notice here we can get a sense of why it gets its name. Here it is connecting to the anterior surface of the ribs 1 through 8, each individual rib. So obviously they're on the front of the ribs, check, anterior. And notice it gives it a scalloped appearance, kind of like a serrated blade, like a steak knife, which is serrated. So serratus anterior. It is anterior in its origin, and its origin has this serrated appearance. Now notice one other thing as well. To see most of this, we had to cut the pectoralis major and remove it, because these are relatively deep muscles. But the key word there is most. Notice in the intact musculature, a couple of those fingers of the serrations can actually be seen. Now, the other picture I really like is this one from your textbook, where again, we see the serratus anterior on the left and on the right. And notice here on the left, they've removed most of the other muscles. So again, we see that origin really nicely here. And I guess I should be doing this in red, but the red doesn't stand out on the red muscles. So there are the origins of it there. And we'll draw it on the skeleton in just a minute. But what is the insertion of the serratus anterior?
Anterior surface of medial border of scapula.
Notice these muscles wrap around the ribs and then attach to the scapula, but they attach to the scapula on the medial border of the scapula. We can draw it on our bones, and we're going to do that in a second. But where I think we can actually see this a little bit better is if we actually draw it for ourselves. So here, for instance, if we were to look at a transverse view, there's your rib cage. Here, of course, is your vertebrae. And let's give it a spinous process because that's fun. Excellent. And then here, of course, is your sternum. And then along the sides here, we have the scapulus. All right. So this is a transverse view of our skeleton. And as we know, the origin of our serratus anterior, the head of it connects to the anterior surface of the rib. But as we just mentioned, the insertion is on the anterior medial border. So it actually connects to the scapula on the anterior side, right here on the medial border. So what this muscle actually does is it wraps around the ribs, not touching the ribs, wraps under the scapula, not touching the scapula, and finally doesn't attach until it gets to the medial border of the scapula. So it's this nice curved muscle that wraps around the ribs underneath the scapula to finally attach to the medial part. So this is our serratus anterior. Wow, that's huge. All right. Now, while we're here, we'll go to our drawings. I encourage you to do this little drawing next to the bones. We'll go to the bones in just a second. But before I do that, there's one other thing I want to remind you about. The goal of a muscle is to pull the insertion towards the origin. So what is the actions of the serratus anterior?
Protract, abduct border, and hold scapula against chest wall.
Right. To bring forward is to protract. Right? You bring this forward. You're giving someone a big hug and you can feel your scapulas. Its job is to hold the scapula in place, stabilize it so that our shoulder joint works properly. However, as we've also talked about, these pesky ribs are in the way. So to bring the scapula forward, I have to wrap it around the ribs as well. So protracting the scapula, remember, we could also refer to as a B. Abducting the scapula. And as we also mentioned, the other action is to twofold. Hold the scapula against the ribs, stabilizing it, holding in place to hold the shoulder in place. But notice also, basically, it can squeeze the ribs as well. And so thereby, by squeezing the ribs, this would play a role in decreasing the volume of the thoracic cavity, which would increase the pressure and force air out. So this is an accessory muscle that helps us to exhale. So that stabilizing or squeezing the ribs is also a function that can help us to help in the exhalation as we're forcing air out. So yeah, the pesky ribs are in the way. It's kind of really got to go to the side rather than just straight forward, but it can still squeeze it, force the air out, stabilizing the scapula, and bringing the scapula forward. Now, I'm going to leave this picture here because we're going to use it for something else in just a minute. But I want to come back to here so we can actually, where is it? There's the Serratus Anterior. The Serratus Anterior here. And again, this is one of those where it's not the bestest of drawings, so you get to be somewhat subjective in how you want to do this. What was the origin again?
Ribs 1 through 8.
Yep, anterior surface of ribs 1 through 8. Excellent. And it inserts into the medial border. Now notice we don't see the medial border. So again, this is one of those things where you can do whatever makes it easier for you. You can draw your insertion on the most medial part that you can see, or if you're able to visualize this 3D for yourself, all right, you can maybe a little lighter or in a dotted line, remind yourself that it is going to be along the medial border of the scapula. So again, whatever helps you to understand where that's located, or like I said, just do your drawing here on the side and stick it right there so that you can see that as well. Whatever helps you to understand it. Again, you're not turning these in, so it's what makes you happy, makes sense to you. All right, questions on that? Now, of course, this is a relatively or mostly deep muscle, but again, there's that pesky word, mostly deep. And remember, on our bodybuilders, we're responsible for superficial muscles, but because a little bit of these peek out, is this something we can actually see on our bodybuilders?
Yeah.
Yeah. Notice here our muscle chart shows it nicely here without that pesky skin in the way. But guess what? If we get a bodybuilder that basically does that exact same pose, lo and behold, we can actually see some of the fingers of that Serratus Anterior there in that picture. And again, I just want you to know that they just photoshopped his head onto my body. That is exactly what my body looks like. All right, questions on that? All right, excellent.
Professor, on the printout that we're putting the markings on, right next to it, the brachialis, brachial, it shows the scapula. Could you just kind of like point out where it is?
Oh, I see. Excellent. Yes. So if you wanted to do, that's a great idea as well. Yes. If you wanted to, like with an arrow, point where it is, remember, it's going to be on the medial anterior border. So on the medial anterior side. Of course, the other way you could do this, do I have it? I don't. Let's cheat. Where would I find it? I would find it here. Again, it's what helps you to understand this. So if you happen to have, because again, as we'll see, a ton of stuff attaches to the scapula. Is there likely to be a scapula on this lab exam? Absolutely. So I could have a scapula and I could have an arrow, right, pointing here to the anterior medial border and ask you what muscle inserts into this bone feature, and what would your answer to that be?
Serratus Anterior.
All right. Or I could take that same arrow and instead point it to the spine and ask you to identify the muscle that inserts into that, and what would your answer to that be?
Trapezius.
Trapezius, there you go. All right, so exactly. So yeah, this, let's paint it. This medial anterior border, that is where the serratus anterior attaches. Okay, great question. Any others? Speaking of muscles that connect to the scapula and are relatively deep, let's cheat and go back a picture. Notice here, oh, I'm not looking, can you see this? I don't think you guys can see this, hold on. That's what I want. There we go. So notice here it's been cut right here. So when we look on the next picture, excellent, underneath the pectoralis major is the pectoralis minor. And here we see that pectoralis minor right here. Now notice I see pectoralis minor here on the list, but I don't see pectoralis major on this list. Does that mean you're not responsible for the pectoralis major?
Yes.
Yes, that would be hopeful thinking. But do you think you are going to be responsible for the pectoralis major?
Yes.
So why isn't it here? Why isn't it on this page of the list that you're looking at?
The upper extremity.
Yeah, because the pectoralis major affects the arm. It moves the arm through space. So when we look at the muscles that move the upper limb, that's where we're going to find the pectoralis major. The pectoralis minor is right under it, but has different origins and different insertions. And notice, as we'll see when we draw the origins and insertions, it doesn't connect to the arm. So it doesn't affect the arm. Instead, it affects the shoulder. It affects the ribs. It affects the trunk of the body. So let's do that. What are the origins and the insertions of the pectoralis minor? Where is the pectoralis minor? It's not on this page. Major's there. There it is. Oops. I'm going to cheat again for my drawing because it's easier for me to get this one up here at the top. So I'll draw it here. You obviously draw it in the right place. So what is the origin of the pectoralis major?
Anterior surface of ribs 3 to 5.
2, 3, 4, and 5. Excellent. And what is its insertion?
Coracoid process of scapula.
There you go. Remember, the coracoid process is that bird's beak right there. So there you go. It inserts right there. Excellent. Notice it doesn't attach to the humerus, doesn't move the arm. And as we know, when muscles contract, they bring the insertion towards the origin. So notice this is going to bring the scapula down. Of course, we're not going to say down. What would we say?
Protract.
What? Well, we'll say
Depress.
To depress, absolutely. Depresses the scapula, brings the scapula down. Notice it also brings the scapula forward or brings it towards the outside. So again, that would be right to protract, stick out, or abduct. And as we've also talked about, we could stabilize the shoulder with other muscles so that when I contracted this, instead of the shoulder coming down, the ribs would come up. So it's going to help us with that inhalation. So bring the shoulder down, bring the shoulder forward, bring the ribs up. Of course, that's not how you're going to describe it on the exam. You'll use the appropriate terms of depressing the scapula, protracting or abducting the scapula, and elevating the ribs. So those are the three actions of the pectoralis minor. Again, remember, don't get confused by where I've drawn this. We're not on the major, we're on the minor. It's easier with the way I've got my picture to use the top one here. Questions on that? This is another one of those that's a little bit tricky. Anybody figure out a good activity, a good mnemonic to help you to understand what this one does? Everybody's silent. It's all right. I get it. I understand it. This one is challenging. My sister says she uses her pectoralis minors to get out of speeding tickets all the time. But I tried it. I got pulled over the other day and I depressed my scapulas, protracted my scapulas, and elevated my rib cage, and I still got the ticket. So I don't know what I'm doing that she's, or I'm not doing that she's doing, but she says it gets her out of tickets. So I don't know. Whatever works for you. All right. Questions on that? I think I pulled a muscle doing that. All right. Excellent. All right. Nothing, not even a laugh. All right, screw all you people. All right, excellent. Let's go ahead. As I've said many times, I tell the jokes for me. If nobody else laughs, I don't care. So
I laughed.
Okay, good.
I was just muted.
That's all right. Excellent.
It's kind of like when you're single and you walk through the bar and you're trying to pick up on ladies and you puff up your chest and
there you go. Exactly. Of course, I'm married. I don't have a chest to puff up anymore. So, but I get your point. All right.
That's why you got the ticket.
Exactly. Excellent. All righty. So let's talk about our next muscle group. Again, notice this muscle group is made up of two muscles, the rhomboid major and the rhomboid minor. And again, do we care?
No, because it's a muscle group.
Exactly. It's a muscle group. And so for this muscle group, we are just knowing them collectively as the rhomboids. And so for the rhomboids, we can, and where is it on this one? Perfect. Excellent. Just don't have to worry about the individual muscles, just need to know them collectively. So collectively for the rhomboids, what is the origin?
Spinous processes of C7 to T5.
Okay, C7, T1, T2, T3, T4, and T5. Excellent. And what is its origin? Pardon me, its insertion.
Posterior medial border of scapula.
Posterior medial border of the scapula. I know I've just drawn this here, but let's go back to our other picture. Notice here we had our serratus anterior. Now what we can do is we can draw our rhomboids. Our rhomboids, and let's use a slightly different color, so we'll use the pink, originates from the spinous process and inserts onto the posterior medial border of the scapula. So this muscle basically connects this way and straight across. So when it contracts, it pulls the insertion towards the origin, which basically brings the scapula towards the midline, which we know is the same as bringing the scapula back. So what is the action of this muscle?
Retract scapula.
Yeah, retract the scapula. Notice if you retract both of them at the same time, like you're doing a rowing motion, you can feel these rhomboid muscles bunching up in between your shoulder blades, right? Because as you're bringing them both back at the same time, you can feel those muscles bunching up along the midline. So that is what this muscle is. So we can see that relationship there. And again, to make sure we understand it, notice here on the exam, I could have these pictures. And if I have the arrow that points here and ask you to identify the muscle that inserts into the this bone feature, what was your answer to that again?
Serratus Anterior.
Yep. But if instead I take that arrow and point to the posterior medial border, now what's suddenly the answer?
Rhomboids.
There you go. See how fun that is? Excellent. So again, if we come back here, we see, and again, here we see it really nicely, right? We have this origin here. We have this insertion here. And as we know, insertions get pulled towards the origins. We're bringing the scapula back. Retract it or bringing it towards the midline. All righty. Now notice one other thing. This is a deep muscle, right? This is a superior, posterior, and deep muscle, right? Notice when the trapezius is in place, we don't see the rhomboids. All right. Questions on that? I think that's all the muscles we need to know the origins and the insertions of, right? For the trunk?
Yeah.
Excellent. But let's do a couple more muscles. We're done with the origins and insertions. Excellent. That's done and out of the way. But let's talk about some of the other muscles. One of the very important muscles that we are responsible for is the diaphragm. The diaphragm is a bell-shaped muscle, meaning that when it is in its relaxed state, it is this curved bell-shaped muscle. We've actually talked about the diaphragm a fair amount in this class. However, up till now, we've primarily just been talking about it as the anatomical structure that divides the thoracic cavity from the abdominal pelvic cavity. And it is indeed that, but it's also a muscle and a bell-shaped muscle. And that's important because when a bell-shaped muscle contracts, what does that bell-shaped muscle do? What happens to a bell-shaped muscle when it contracts?
Flattens.
Yeah, basically all muscles get shorter when they contract. And when a curved muscle contracts and gets shorter, it flattens out. This is important because, as we know, pressure is what makes the world go round. And when this bell-shaped muscle flattens, the volume of the thoracic cavity increases. And what happens to the pressure when volume increases?
Also increases.
Yeah, no. Pressure decreases. Remember, when we're talking, especially when we're talking about gases, but really when we're talking about everything, pressure is basically a measure of the things that bounce against the walls. Again, I have to work on my analogies here because usually we're in the classroom. So the example that I use is we're all sitting here in the classroom right now. I blindfold all of you and I allow you to walk around the classroom. And as you guys randomly walk around the classroom, every once in a while, one of you is going to bump into the wall. And basically, I count the number of times someone bounces into the wall for a minute, and that gives us our pressure. If I then brought all of you here and put you in my bathroom, not surprisingly, my bathroom has a much smaller volume than our classroom does, right? What would happen to the number of times you guys bounced into the walls?
Increase.
It would increase. So when volume goes down, pressure increases. Conversely, if I released you from the classroom and let you roam around campus, and I counted the number of times you guys bumped into the boundaries of the campus, when the volume goes up, the pressure goes down. And so that's what happens here. When our muscle contracts, again, this is the contraction, this is the active process. And of course, what do I mean when I say it's an active process?
You control it.
True, but that's not necessarily what I mean. That is true. How do I control it? What makes it active?
ATP.
Yeah, uses ATP. Excellent. So when we contract, that active process of using ATP, it lowers the pressure. And as we know, the thoracic cavity just happens to be open to the outside world by your respiratory system. So when the pressure decreases inside of here, air moves in. So notice when we're actively using ATP, we are actively using it for the active act of inhalation or inspiration. Of course, we don't leave that muscle contracted forever. Eventually, we will relax the muscle. Of course, relaxing the muscle is a passive process. Does not use ATP, but it does use one of the passive properties of muscle tissue. And we know muscle tissue, when the pressure is removed, when the force is removed, is elastic. It has extensibility and elasticity. And so when we relax the pressure, relax the muscle, it goes back to its original shape. And when it goes back to the original shape, what happens to the volume of the thoracic cavity?
Decrease.
It decreases. And when the volume decreases, what happens to the pressure?
Increases.
And because this is open to the outside world, thanks to the respiratory system, when that pressure increases, air moves out. And we exhale. Expire. Our diaphragm is the primary muscle involved in your normal resting breath. What is called tidal breath. So again, while you're just sitting here, calmly listening to me, breathing through your nose, taking small, of course now yawn, taking small normal breaths. That normal tidal breathing is primarily controlled by the diaphragm. And notice it does both. The diaphragm is responsible for inhalation and exhalation. However, what is its action? What is the active process that it actually is involved in? When it uses ATP, what does it do?
Increase thoracic cavity.
Yeah, brings air in. So when we are actively using, when you actually contract the muscle, you're bringing the air in. Air leaving is just passive. Now, as we've talked about, are there muscles that can help us to take an exaggerated inhalation?
Yeah.
And an exaggerated exhalation?
Yep.
Yeah. In fact, we've already talked about some. What are some of the accessory muscles we've talked about that can help us with an inhalation? I can think of three we've talked about.
Serratus Anterior.
Now, Serratus Anterior was an accessory muscle, but remember, that's the one that squeezes the thoracic cavity and forces the air out. So that one would actually be our exhalation.
Trapezius.
Trapezius, remember, didn't affect the ribs so much, although there was an inhalation muscle we did talk about today. Brings the chest up.
Pectoralis Minor.
And there were two muscles we talked about in the last class. Neck muscles that also could bring the ribs up.
Scaling.
Excellent. And then the alphabet soup one, which one was that?
Sternocleidomastoid.
Excellent. There are a couple more we'll add to this list, but this is a good starting point. So notice we have accessory muscles that are useful for this process, but the normal resting tidal breath is almost entirely just the diaphragm. But when we need to take a bigger inhalation or a bigger exhalation, we have other muscles that can help. Notice you see this muscle really nicely in your illustration from the textbook, this nice big curved bell-shaped muscle. Notice attached to the ribs. We don't need to worry about its origin and insertion, but it forms that anatomical barrier. And again, notice there are some things that have to sneak through. So notice there's an opening for some blood vessels like the vena cava and the aorta. Your esophagus, some nerves have to get through here, stuff like that. But for the most part, it forms that important barrier. And supporting the thoracic cavity and changing the volume, which changes the pressure that allows us to breathe. Now, this muscle is the diaphragm, but it turns out we have another diaphragm, a urogenital diaphragm. The urogenital diaphragm, just like your diaphragm supports the organs of the thoracic cavity, urogenital diaphragm supports the organs of the pelvic cavity. Here we see the female version, but males have it as well. There are numerous muscles involved in this, but the primary muscle of the urogenital diaphragm, so again, we're talking about the urogenital, is a muscle group called the levator ani muscle. The levator ani muscle is made up of two muscles, the pubococcygeus and the iliococcygeus. And while those are incredibly fun to say and spell, do you actually have to know iliococcygeus and pubococcygeus?
No.
No, you just need to know them collectively as the levator ani muscle. As we can see, this urogenital diaphragm, as we talked about, helps to support the organs of the pelvic region. And as you also see, this is going to play a support role in both defecation, helping to elevate the anus, in helping of the expulsion of the feces during defecation, and in micturition, which of course is the process of voiding urine from the body. I know most people say urination, but if you think about what that term means, urination means to make urine. You're actually making urine all the time. Every single person in this class right now is urinating as we speak. Your kidneys are filtering the blood and producing urine as a result. But hopefully urine isn't constantly leaking out of your body. Instead, it is going down into your bladder where it is stored till you can get to a socially appropriate location and void the urine at that point. And that process of voiding the urine is actually micturition. Right? If you have ever been at a bar on a Friday night, had one too many Long Island iced teas, right, and needed to use the restroom and saw that the line for the restroom was 75 people deep, which is silly because there's some perfectly good bushes just out back. So there you are out in the bushes behind the bar, you're micturating, and the flashlight from the cop lands on you. Do you continue to micturate at that point?
No.
No, you stop midstream, pull up your pants, and run as quickly as you can. That contraction that you make to stop urine mid-flow, that is that contraction of that levator ani muscle. Who else are these contractions of the levator ani muscle important for? Maybe it helps
Women.
if I do that. Exactly. Because not only do women get to use the levator ani to help in defecation, to help in urination, but it also supports their reproductive structure, their vaginal canal. Believe it or not, passing a basketball out of your vaginal canal is a fairly traumatic process for the urogenital diaphragm and can weaken the muscular structure of a female. It's not uncommon for a female who is recently given birth. If she laughs too hard or sneezes, what can happen as a result of that?
Ankle.
Yeah, a little bit of urine could sneak out, right? Because of that weakening of the muscle. So one of the things that pregnant women often do near the late stages of their pregnancy is do a special exercise where they make that contraction like you're stopping the urine mid-flow, hold it for 10 seconds and let it go, do it in sets of 10. And the advantage of it is you can do it all the time while you're sitting at a red light, standing in line at Starbucks. I've just done 4 of them sitting here, right? And what do we call those exercises?
Kegels.
Kegels, absolutely. Yes, it's important so that the girl, the woman doesn't urinate when she laughs. But even more importantly than that, with a weakening of the urogenital diaphragm, what actually can happen is the vaginal canal can actually descend out of the urogenital diaphragm. It's a condition, they call it prolapsed vaginal canal when that occurs, and actually has to be sometimes fixed surgically where it's ascended and put back into place. So strengthening those levator ani muscles with those Kegel exercises are very, very important for females during pregnancy. Recent studies have also shown Kegel exercises can help males. Kegel exercises in males have been shown to prolong time before ejaculation takes place. So it can extend the duration of intercourse for the males, which is beneficial for the females as well. All right. So there you go. 2 actual diaphragms, our diaphragm, and then this urogenital diaphragm made up of the levator ani muscle primarily. Questions on that? Again, we don't need to know the origins and insertions, but obviously the location of it is fairly obvious here in the pubic region. All right, questions on that? Oh, and there you go. Here we see the levator ani side by side in males and females. Perfect. Okay, excellent. Perfect. This is what I wanted to do. All right, questions on that before we move on? All right, we've been talking about our respiratory muscles, and that brings us to our intercostals. Our intercostals are associated with our ribs. So let's draw a couple ribs here. Close enough. Obviously, we also need our sternum here in the middle. Up and I'm gonna have to cheat a little bit. Move this way, that, that. There we go. And of course, as we know, a little better. Yeah, that looks pretty decent. Our ribs are connected to the sternum by cartilage. Excellent. So what we are looking at now are the intercostal muscles. And as our handout and everything else shows, our intercostals come in two flavors. They are the external intercostals and the internal intercostals. Now, based on their names, and I'm going to use, actually, I won't use red and blue because I don't want to be confusing. We'll use brown and orange. Or brown and green. No, I used green already. Brown and some other color. Based on their names, what do you think one of the big differences between the external intercostals and the internal intercostals are?
Location on the ribs.
Yeah, absolutely.
Outside of ribs, inside of ribs.
Close. They're intercostals, so they're between, but in between the ribs, there are two layers of muscle. One is deep and one is superficial. So which of our two intercostal muscles here do you think are superficial?
External.
There you go.
Intercostal.
And then obviously our internals are going to be deep. But there are two other big differences between our internal intercostals and our external intercostals. Our internal intercostals are indeed deep, but they're also more medial. These actually touch the cartilage and the sternum. Whereas the external intercostals are going to be more lateral and won't get all the way to the cartilage. And then the other big difference is the orientation of the fascicles. For our internal intercostals, the fascicles on our internal intercostals go down and out, meaning they project inferiorly and lateral. So if we were to draw these, they start here medially, and the fascicles go down and out. Now notice the same thing happens on the other side as well. They're going to go down and out on the other side as well. So they are medial, they are deep, and their fascicles project down and out. Oops. All righty. Our external intercostals, as we mentioned, are the superficial layer, but they are more lateral. Do not reach the cartilage. And their fascicles project down and in, meaning that they project inferior and medial. So they are going to start more lateral, and they're going to project down and in. Now they do overlap, but remember, they're not going to get to the level of the cartilage. Same thing on this side as well. They are going to project down and in, overlapping, being on top of the deep internal intercostals, and project down and in. All right. Questions on that? Again, we don't need to know the origins and insertions of these, but we do need to be able to recognize and identify them. And this is going to way we're going to do them. And we also need to know their actions. Now, as we mentioned, our intercostal muscles are accessory respiratory muscles. And of course, our two respiratory processes are inhalation, that begins with an I, and exhalation, that begins with an E, which is awesome, because then it would be really, really convenient if our internal intercostals inhaled and our external intercostals exhaled. And is that what happens?
No.
No. It would be very convenient, but it's not the case. Our internal intercostals are the ones that actually bring the ribs down. And so as a result of that, they're responsible for exhalation, whereas our external intercostals bring the ribs up. And so they're responsible for inhalation. So our externals inhale, our internals exhale. It's not perfect, but at least while I, E, E would be nice, we can just remember that E goes with I and I goes with E. So almost as good as a mnemonic. Now, of course, we've done this simple illustration to look at this, but when we look at the pictures, we see the exact same thing. Notice superficial, lateral, and fascicles coming down and in. We have my external intercostals. Notice deep, medial, touching the sternum, touching the cartilage, and fascicles going down and, well, that orange doesn't show up at all. Going down and out are the internal intercostals. And what I really like, your book's got this great picture that conveniently enough peels the two layers apart. Obviously, when we peel the two layers apart, we can't see which one's superficial and which one's deep. But we can clearly see that the external intercostals don't come up to the cartilage, don't come up to the sternum, whereas the internal does. And we see the fascicles here are coming down and in, whereas the fascicles here are going down and out. So again, when they separate them, it makes it really easy to tell which is which. We can't tell which is superficial and deep, but we have two other factors to tell which is which. All right. Questions on that? All right, excellent. All right, that brings us, how are we on time? think we can sneak this in. Let's do this. All right. Again, I want to start here. I'm going to actually save this picture because I want us to understand one other important thing to remember as we move on to the next part. As we move on to the next part, I want you to remember how the internal intercostals, their fascicles went down and out, and the external intercostals, their fascicles went down and in. Okay? All right, with that in our pocket to save for later, we are going to now talk about a muscle group known as the abdominal girdle. What do you think the function of the abdominal girdle is?
To stabilize the organs in your abdomen.
Absolutely. We've got a whole bunch of organs here in our abdominal pelvic cavity, and very few of them have bones that protect them. Because of the diaphragm, there's a couple organs that get to sneak up a little bit underneath the ribs. But the vast majority of the organs in the abdominal pelvic cavity are not protected by any bones. So it's muscles, and muscles at lots of different orientations. If we're going to have layers of organs, lots of different layers of these muscles provide protection. And in fact, we essentially have 4 layers at 4 orientations. Let's start easy. One of the sets of muscles that we have in our abdominal girdle are a set of muscles that run vertically. And there's like like many muscles, they're paired. We have them on both sides. So we have these long, straight, vertical muscles. What are these long, straight, vertical muscles called?
Parallel muscles?
This is a parallel muscle arrangement, but again, as we mentioned, muscles also often get their name by their shape. A muscle that is long and straight is often given the name rectus. This rectus muscle, long straight muscle, happens to be in the abdominal region. So guess what they call this rectus muscle?
Rectus abdominis.
There you go. Bingo. Rectus abdominis. Now, you may not have recognized my drawing of it because one of the interesting things that occurs with our rectus abdominis is there are actually small tendons, and those tendons actually divide this straight rectus muscle into not 1, not 2, not 3, but 4 individual bellies on each side, which, if you happen to have the tone and the lack of adipose, these can be clearly defined, and we commonly refer to them as what?
6-pack.
Your 6-pack. Your 6-pack, first of all, is actually an 8-pack. But absolutely, your 6-pack is those tendinous insertions that isolate each of these individual bellies of our rectus abdominis. What's really interesting about them is each of these 8 bellies are actually independently innervated, meaning it's possible to just contract one of these bellies. Now, just because it's possible doesn't mean that everyone can do it. Just like everybody has levator muscles on their ears, and yet is everybody able to wiggle their ears?
No.
Anybody here able to wiggle their ears, at least willing to demonstrate, or at least claim to be able to do it?
I can't.
No, but if you and Elvina can't, then nobody else is going to speak up. So we'll just assume somebody can and they're really, really shy. So yeah, again, just because some people, we all have the muscle to allow us, it doesn't mean we have the control. But there are plenty of videos. Terry Crews is probably one of the most famous ones who actually are able to individually control these. There's a great video on YouTube where he has all these electrodes connected to the muscles of his body. And as he contracts the muscles and they generate an electrical current, that current actually stimulates a musical instrument. So he basically plays a whole orchestra just by contracting muscles of his body. So there are people who have that type of precise control. We all are capable of it, but we don't necessarily all have it. All right. Now, the other thing to remember when we're talking about muscles is to think in terms of how they affect the bones. And in this case, the bones that are affected by all of the abdominal muscles are the vertebrae. So we have the vertebrae, our thoracic and lumbar vertebrae, and it is those vertebrae that they influence and affect. So of course, when our abdominal girdle contracts, those muscles get smaller. They basically move parallel to the vertebral column. So what does that do to the vertebrae? What is the action?
Flexes the vertebrae.
It flexes the vertebrae. Now, and as we've also talked about, this abdominal girdle's job is protection. So when we engage these abdominal muscles, engage your core, it also constricts the size of the abdominal pelvic cavity, strengthening these muscles. So the other thing that it does is it compresses. The abdomen, right? So that when someone's getting ready to punch you in the belly, or you're walking on the beach and that Jonas brother walks by, right? You suck in the gut, compress the abdomen so that you look a little bit more impressive. So those are the actions. All of our muscles are going to play a role in compressing the abdomen because, again, their job is that protection. These are the most medial of our abdominal girdle muscles. And not surprisingly, if we have muscles that are vertical, we also have muscles that are horizontal. These horizontal muscles are the deepest of our abdominal girdle muscles. And what are these deep horizontal muscles called?
Obliques.
Transversus.
There we go. Like
abdominis.
Exactly. Like if you were taking a cross section of the body, this would be a transverse section. So it's the transversus abdominis.
Is it NUS or NIS?
US is the singular, IS is the plural. I don't care either way. And again, you get one letter just like I do. Excellent. Now, again, deep muscles, horizontal in their orientation. We also want to think of these muscles in terms of their actions. If you think about this muscle, because it's horizontal, and I'll use my pen here, basically comes around the body in a big circle and back to the other side. Notice it doesn't have a way to really interact between vertebrae. Essentially, what it forms is a circular muscle. And as we know, when a circular muscle contracts, that circular muscle gets smaller. So really, there's only one action for the transversus abdominis, and that's to compress the abdomen. Excellent. So we've got horizontal, we've got vertical, and we have angles. Angled muscles are easy because since they're angled, how are we going to refer to them?
Internal and external.
Internal and external what?
Oh,
What is it?
Oblique.
Oblique, right? Remember, oblique means at an angle. So you're right. We are going to have an internal and external oblique. And notice they're perpendicular to each other, kind of like we saw with our intercostals. And notice our red oblique here, its fascicle projects down and in. So which oblique do you think this would be? Which
Internal.
intercostal? Yeah, well, which intercostal projected down and in?
Oh, external.
External. So lo and behold, our external oblique projects down and in. Wow, did all sorts of things wrong on this. That's right. Now remember, the way the fascicles project is only one of the ways we're going to tell them. Obviously, being the external oblique, it is also going to be more superficial. It's going to be that superficial layer that is going to be on top. But there's one other way that our external oblique is different. It is more superior. It actually goes up above the ribs. In fact, it actually interdigitates with the serratus anterior. What does it mean to interdigitate?
Connect.
Yeah, basically just interlocks with. So the external oblique comes up to where the serratus anterior are, and they basically just, like your fingers, interdigitate, overlap, interlock, I guess would be another way of saying that. And their fascicles go down and in. So again, on this side, that's this way. On this side, that's that way. So our internal oblique muscle, like the internal intercostals, have fascicles that project down and out. As the name indicates, it's the deep layer. And there's a third way that they're different, more inferior. Only come up to the ribs. They do not go over them. So they are, again, down and out, down and out, down and out, down and out. And so we have all these interlocking layers of muscles. Now, notice the one thing we haven't done. We haven't done the actions for the internal oblique and the external oblique. When you look at your list, what do you notice about the actions?
are the same.
Absolutely are the same. And if you look closely at this picture, you can kind of see why that would be. Notice the orientation of the internal oblique on one side of the body is the same as the orientation of the external oblique on the other. So when you move your core, when you move your trunk, you're basically using the external on one side and the internal on the other. Which, since they're red and blue, conveniently I'm writing here in purple down below. And what are the four actions? Oops. The four actions of our internal and external oblique?
Compress abdomen.
Right, okay, excellent. So compress abdomen. So again, as we know, that means bringing the gut in, right? Tightening up the muscle when someone's getting ready to punch you. What else?
Legs, rotate the roll column.
Excellent. So again, flexing would be bending forward. What else?
Lateral flex.
Lateral flex, bending to the side. And what's the last one?
Rotate.
Rotate. There you go. And those actions are the same for both the external and the internal obliques, because when you do one of those things, like twisting to the side, you are pulling with the internal on one side and the external on the other. Now, I like this simple illustration because I think it does a good job of showing us what we need, but we definitely need to see it in the body. Notice this picture does a good job of showing this. So notice here we have our most superficial muscle with its fascicles going down and in, and notice it comes up and interlocks with the serratus anterior. So this is our external oblique. But notice on this side, they've cut it. So we see the internal oblique that only comes up to the ribs, and its fascicles go down and out. And notice they've also made an additional incision where we can see the horizontal muscles of that deep transverse abdominis, deep here. These three muscles all overlap to the side, but notice here on the midline, or close to the midline, we can see that long straight muscle that is that long straight rectus abdominis with its tendinous insertions to give us those individual bellies. Useful, but we have some better pictures as well. Get to that in a second. Here's the one I like the most. Actually, that's not true. This is the one I like second most. Again, they've done a good job of peeling apart the individual muscles for us. Notice external obliques, their fascicles come down and in. Because we've separated it, we can't tell it's the most superficial, even though we know it is. But notice we can clearly see that it is coming up over the ribs to where the serratus anteriors would be located. Whereas notice the internal oblique doesn't really go over the ribs, doesn't get to the serratus anterior, and its fascicles go down and out. Our other two are more straightforward. Vertical rectus abdominis, horizontal transversus abdominis. But another place where we see this really nicely is in a cross section through the body. Notice here we kind of get a couple of bits of their relationship to each other. We see medially those rectus abdominis muscles that we've cut in cross section. And then out here, we see not one, not two, but 3 layers of overlapping muscle. External oblique is the most superficial. Deep to that is the internal oblique. And the deepest is the transversus abdominis. So both of these 2 pictures, this one and the previous picture, do a really great job of showing the relationship of these muscles isolated and to each other. But there's really two other things that this picture shows really, really nicely. Notice that this picture also does a really good job of showing us how each of these muscles have a big, broad, flat tendon that come off of them. What would we call a big, broad, flat tendon that comes off of these muscles?
Neurosis.
Yeah, if you're not sure, it says it right here. Absolutely, aponeurosis. Notice all of them have them. But when we're looking at an intact abdomen, how many of them do we see? Just the one, just the aponeurosis of the external oblique. So if we go back a couple pictures, well, this one showed it nicely. Here we see the aponeurosis of the external oblique. Yeah, let's go ahead and erase all that. Aponeurosis of the external oblique there. But the other place we see it really nicely is here. Notice they've kind of given us this shaded part here where they've shown us the aponeurosis of the external oblique. Now we know all of them are there, but we're only seeing the aponeurosis of the external oblique, which happens to be one of the structures you are responsible for on the exam. But the other thing that picture showed us is that the aponeuroses from all of these muscles come together to form this midline white band of collagenous connective tissue that runs right down the center of the midline of the body, right through the belly button. And of course, this was famously named after Jessica Alba's younger sister. So what did they name this structure?
Linea alba.
Linea alba. Alba, of course, means white. Linea means line. It's the white line. Now, you are not responsible for the origins and the insertions of the abdominal muscles. But one of the things that I do need to point out to you is that there is a very important ligament down here. This ligament attaches to the superior anterior iliac spine of the pelvis and goes to the pubic tubercle. And this ligament is what is known as the inguinal ligament. Let's jump ahead a picture. No, 2 pictures. Notice this picture shows it here and here and here from the superior anterior iliac spine to the pubic tubercle. So notice all of the abdominal muscles attached to it, but there's lots of other muscles that attach to it as well. And the reason I mention it is twofold. One, because it is a structure you're responsible for on the exam. So that right there is a good enough reason to do that. But let's go back to this picture. Notice over here, there is a space or potential space that passes through it. In males, this is an actual space called the inguinal canal. And as you can see, the spermatocord which is the fibrous structure that brings the blood vessels to and from the testes, the nerves to and from the testes, and the sperm from the testes into the abdominal pelvic cavity, passes through that opening into the abdominal pelvic cavity, right? Because the testes are located outside the abdominal pelvic cavity for temperature regulation and so that you can win $10,000 on America's Funniest Home Videos. So it has that opening that it has to pass through to get inside the abdominal pelvic cavity. Females have an inguinal canal as well, but for females, it's a potential space. I think we've talked about this before in this class, but just if in case we didn't, we'll do it again. Notice when I put my two hands together, is there anything between my hands? No, not really. But does that mean my two hands have fused together to form one structure?
No.
No.
No, there is a potential space there. I can make a space between my hands, but when I slap them together, there's no space there. It's just a potential space. And that's what it is in females. In females, it is a potential space. In males, it's an actual space. This is important because, as we know, pressure makes the world go round. And if you were to increase the pressure inside your abdominal pelvic cavity too much, what can actually happen is some of the fluid, peritoneal fluid, some of the adipose, and even some of the organs could actually bulge out of this opening. This opening is basically a weakness into the abdominal pelvic cavity. So if you're trying to lift something that is too heavy, or catch something that is too heavy, or exert yourself too vigorously while defecating, right? About a third of these actually happen via defecation. What do we call that condition when something expulses itself out of that opening?
Hernia.
Yeah, and in particular, this is an inguinal hernia. Inguinal hernias are far more common in males than they are in females, because in males, it's an actual space, and in females, it's just a potential space. There is one other weak point that is equally likely to have both males and females get that hernia to occur. And what is that additional weak spot that both males and females have? Only there was a weakness inside of our abdominal pelvic cavity that everybody...
Belly button.
Belly button, the umbilical hernia, absolutely. The umbilicus is another weak point in the wall of the abdominal pelvic cavity where you can get that expulsion. And since both males and females equally have belly buttons, they're both equally likely to get an inguinal hernia. Excellent. One more structure on your list you are responsible for, and that's this one on the back. Notice this is the diamond-shaped white structure here on the posterior part of the body. The name helps us in that it is the thoracolumbar, meaning that it basically is in the thoracic and lumbar region of the posterior part of the body. But notice it's not an aponeurosis. It is big, broad, flat pieces of collagen fibers, but it isn't just the tendon of one muscle. Several muscles, tendons feed into it, but it's also a protective connective tissue sheath sitting over the many tiny muscles and joints of the vertebral column. And as such, it is therefore a connective tissue fascia. So on the front, we have aponeuroses. Here on the back, we have the thoracolumbar fascia. Questions on that? All right, excellent. Then just that easily, I think we've hit everything on our trunk list. all the muscles, all the actions, all the origins and insertions we're responsible for, and now all of the structures. Awesome. Shockingly, something I didn't think I would say in the summer, we are actually ahead of schedule. We actually got this done, and we're actually ahead of where I usually am at this point. So that is absolutely awesome, which is good. More time, the more material we can cover, the more you have to study, the more time you have to study it. So excellent. This is an awesome, awesome place to be. I am stoked. So I know we went a little long today, although we didn't. We're still in our time. We took advantage of damn near all of it. So I know that makes for a long day, and I apologize and appreciate that. But like I said, the more we can go over this stuff now, the more time you have to digest it and make sense of it. All right. Questions on this or anything else? All right, excellent. Then have a good day, and I will see you guys tomorrow. We get to do 4.5 more hours tomorrow. Yay, summer school. All right, see you guys. Have a good day.
Thank you. Bye.